Good morning. My name is Fotini Panagi, and I have the pleasure of serving as the Master of Ceremony for the opening part of the 15th Annual Scientific Conference of the Center of Applied Neuroscience. Before we begin the conference, we would like to inform you that photography, audio, and video will occur during the event. I would like to acknowledge and welcome Professor Tatiana Eleni Sinodinou, Vice Rector of Academic Affairs of the University of Cyprus, Professor Antonis Selinas, Dean of the Faculty of Social Sciences and Education, Professor Kostas Fandis, Chair of the Department of Psychology, Professor Fofi Konstantinidou, Director of the Center for Applied Neuroscience, members of the FAN Faculty Board, members of the CAN of Advisory Board, and distinguished members of the Department of Psychology, our esteemed invited speaker from Cyprus and abroad, dear delegates and dear students, ladies and gentlemen, good morning once again. I hope I have not left anybody out. The Center of Applied Neuroscience of the University of Cyprus warmly welcomes you to the 15th Annual Scientific Conference entitled Innovations in Integrative Neuroscience, Precision Approaches for Cognitive and Psychological Interventions. We also wish to highlight that this year's conference is supported through funding from the BRAIN project, Brain Research and Integrative Neuroscience Network for COVID-19, a Horizon Europe twinning initiative coordinated by the Center for Applied Neuroscience. We also thank the Department of Psychology for supporting this important conference. A warm welcome to our keynote and invited speakers and to all participants joining us from Cyprus and abroad. We are delighted to have neuroscientists, healthcare professionals, scientists, researchers, and students from various disciplines with us today. This annual scientific conference contributes to the mission of our center, which for 15 years has served as a point of reference and collaboration for the scientific community in Cyprus. At the same time, it supports the dissemination of knowledge on the latest developments in the study of the human brain and the links between biology, cognition, and behavior. We hope you will find today's program engaging, informative, and inspiring. Without further ado, I kindly invite the Vice Rector of Academic Affairs, Professor Tatiana Eleni Sinodinou, for her welcoming address. Mrs. Vice Rector, the floor is yours.
Dear colleagues, distinguished guests, ladies and gentlemen, good morning. It is with great pleasure that I welcome you to the 15th Annual Conference of the Center for Applied Neuroscience. This year's conference, which is organized in collaboration with the Department of Psychology of the University of Cyprus and the Horizon Europe funded research project Brain Research and Integration Neuroscience Network for COVID-19, BRAIN, will focus on innovation in integrative neuroscience, precision approaches for cognitive and psychological interventions, a theme that reflects the Center's commitment to applying innovative approaches to improve mental health and well-being. Allow me to begin by saying a few words about CAN, the organizer of the conference. CAN is an approved research center by the government of Cyprus and the only independent research center in the School of Social Sciences. Its mission is to conduct research that links brain physiology and function with human cognition and behavior in typical and clinical populations. Through its activities, CAN contributes to the development of new knowledge, methods, and technologies that advance science and benefit health, quality of life, and society at large. Building on that mission, over the past decade, CAN has established itself as a major player in neuroscience research, both nationally and internationally. With a funding portfolio of over 30 projects and a cumulative budget of 22 million euros, half of which managed by the University of Cyprus, the center remains largely self-sustainable. The funding has enabled the training of more than 100 postdoctoral fellows, master and PhD students, contributing significantly to research capacity building at the University of Cyprus and in Cyprus. The center's research is multimodal and interdisciplinary and integrates complex methodologies in its quest to tackle and provide answers to mental health and well-being issues. These achievements have allowed CAN to engage in high-level international collaborations. The three advanced partners of CAN to the BRAIN project, the University of Maastricht, King's College London, and Ghent University, have world-renowned expertise in neuroimaging, non-invasive neurostimulation, and digitalized assessment methods, extensive research management experience, and active interest in post-COVID research. Their expertise, combined with the expertise of the CAN team on neuropsychological rehabilitation, generated the cutting-edge research methodologies implemented in the BRAIN project. Equally important are the training and capacity building activities of the project, such as this conference and workshops, which facilitate the transfer of knowledge and expertise to Cyprus researchers, one of the main objectives of the twinning funding mechanism. The establishment of research centers like CAN has created significant momentum for both the University of Cyprus and the society. The university remains committed to working closely with the center's director, the faculty, and the advisory boards to ensure that CAN continues to fulfill its vision and expand its role and position with the neuroscience community, both nationally and internationally. Closing, I would like to thank all those who contributed to the organization of this conference, especially Professor Fofi Konstantinidou, as well our esteemed speakers and participants whose presence, expertise, and engagement will surely make this gathering a success. I am confident that the conference will stimulate great discussions, will offer new opportunities for collaboration. I wish you all a productive, thought-provoking, impactful, and inspiring conference. Thank you for being here, and thank you also for supporting with your presence the centers and the University of Cyprus activities.
Thank you, Professor Synodinou, for your welcome address. Next, I invite the Dean of the Faculty of Social Sciences and Education, Professor Antonis Elinas, to offer his remarks. Professor Elinas, the floor is yours.
Well, good morning, everyone. And on behalf of the School of Social Sciences and Education, a very warm welcome to the 15th annual conference of the Center for Applied Neuroscience. So our school is home to about 2 out of the 7,000 students of the University of Cyprus and to the departments of Education, Law, and Social and Political Sciences, and of course, to the Department of Psychology. Our school is very proud for its devotion of its faculty to teaching, but also for its strengths in research and in its societal outreach and impact. And I would like to say a few things about this latter two, about research excellence and societal impact. So with regards to research, we're very proud that, and the chair of the department is here, the Department of Psychology is one of the top-ranked fields at the University of Cyprus and the top-ranked field in the broader Greek-speaking university world or ecosystem, as we say. And I think this is testimony to the strengths of my colleagues in producing internationally acknowledged research, and we're very proud of that. As a school, what we have tried to do is to further facilitate the department reaching its research potential through the investment in research infrastructure and research equipment. So in the past few years, we have devoted considerable time to make sure that our colleagues have the equipment and infrastructure necessary to do their work. And we are very proud that beginning in 2026, we'll also have the chair of the department, Kostas Fandi, has worked a lot for this, a virtual reality lab with about 40 stations that will further facilitate both teaching and research. And we're very thankful to the vice rector and to the university, the central administration for facilitating this. As a school, because we want to encourage even more research, we also, in the past few days, approved a research incentivization scheme to enhance our presence in top grant applications, for the major funding agencies, including the ERC ones. And finally, we're very thankful that the Central University is helping the Department of Psychology and the school secure a new faculty line in the neurosciences. We expect that this is going to happen very soon, and we're very thankful for this support. Now, CAN is very central to the research mission of our school because as a center, it devotes a lot of attention to the contact of basic and applied research and also to training and employing our doctoral students, our postdocs, our master students, as well as undergraduates. So we're very thankful for all the center is doing to facilitate research. Now, in addition to that, our school is also devoted to having a broader contribution to society. And again, CAN is very central to the broader activities of our university and our school in terms of societal outreach. CAN faculty and researchers have a dynamic presence in the media, in non-governmental organizations, and in advocacy groups. They regularly hold information talks and presentations with the greater public. And overall, they try to bridge these two worlds of the academy and the broader community. Just to give two examples, I think BRAIN is a good example of this great citizenship. So its involvement and the BRAIN team in the assessment and also the development of innovative therapy tools for long COVID patients is an example of this attempt to provide services to patients and to society in general. And it is the only unit that does that in the country. So we're very, very thankful for this. And it's an example, again, of this meaningful societal outreach. But also, a second example is this conference. So this is the 15th annual conference. In the academic world, it's easy to start things, but it's very, very hard to keep them going. And this is the 15th annual conference, and it's open, as always, to the public and to society. And I think it's another example of this attention and focus of CAN, not only on research, but also in making research outputs known to broader society and involving and learning from society as well. So it is very key to our mission, the school mission for both research excellence and citizenship. And here I would like to thank my colleague, Professor Constantinidou, for heading CAN so successfully and steering it in this nice direction of combining both research excellence and societal outreach. I would also like to thank my other colleagues at the Department of Psychology, Timothy Papadopoulos, Georgia Panayotou, George Panoudis, Evangelos Paraskivopoulos, and Panos Zanos. So this is the rest of the faculty which is involved in CAN. You probably know them bibliographically, those coming from abroad, or you know them personally, but probably you don't know how much they also offer to the school and to the university in terms of good citizenship, and I would like to sincerely thank them for that. And of course, I would like to end by extending a warm welcome to our guests. We're very thankful that you're here and supporting this important project. I regret, and I have to apologize, that I have to leave right afterwards because of another not so interesting meeting and gathering. But I'm confident that the conference will stimulate great discussion, and I'm happy to see that there's so many people that have attended. I wish you a very productive session. Thank you.
We thank the Dean. Next, I would like to invite Professor Fofi Konstantinidou, Director of the Center for Applied Neuroscience, for her welcome address. Professor Konstantinidou, the floor is yours.
Good morning, everyone, and welcome. Professor Sinodinou, Vice Rector of the University of Cyprus, Professor Elinas, Dean of the Faculty of Social Sciences and Education, Professor Fandi, Chair of the Department of Psychology, dear members of the CAN Advisory Board, distinguished guests, dear colleagues, dear students, ladies and gentlemen, good morning. It is my great pleasure to welcome you to the 15th Annual Scientific Conference of the Center for Applied Neuroscience, CAN, at the University of Cyprus. Our annual conference is a key dissemination and communication event that help us fulfill our mission. CAN is the leading center dedicated to the study of mind, brain, and human behavior in Cyprus, and our vision is to become a flagship center for research and collaboration in our broader region and beyond. So this year, we're really excited to welcome about 250 participants, one of our largest conferences. Well done, everyone. Thank you for being here. So let's begin with a couple of warm-up questions for the rooms. When was the first CAN conference? What year was it held? It was 2011, September of 2011. How many of you were present at that conference? Yeah, a few of us were present. How many research teams conduct research at CAN at this time? How many would you say? Any other answers? I heard 5. It's 6 research teams. And as I name the teams and the PIs, those of you who are members of those teams or even alumni of those teams, you know, you've graduated and moved on, or you were a postdoc and moved on, please stand. So, Translational Neuropsychopharmacology Lab with Dr. Panos Zanos. Those of you who are part of this lab and your director. Okay. Cognitive Neuroscience and Functional Neuroimaging Lab with Dr. Evangelos Paraskivopoulos. Awesome. Clinical Psychology and Psychophysiology Lab with Professor Georgia Panayotou. If you're here, any of the lab members? Georgia will be joining us later. She's at a School of Graduate Studies meeting. She's the Dean of Graduate School. Let's see. Language and Cognitive Development Research Group with Professor George Spanudis. Anybody from the group here? We're still applauding you guys. Yeah. Let's see. Learning Disabilities Research Group with Professor Timothy Papadopoulos. Anybody from the lab? Timothy is on sabbatical and is currently abroad, but he's sending his regards. And let's see, anybody from my lab, Neurocognitive Research Lab? Yeah, nice. Okay, I didn't miss anybody. I think I got us all. So our teams are committed to excellence and to advancing applied research that addresses real-world needs across cognition, behavior, language, and affect, and brain function. We study individuals across the lifespan, from childhood to older adulthood, covering developmental, psychological, and acquired brain conditions. This year's theme, Innovations in Integrative Neuroscience, Precision Approaches in Cognitive and Psychological Intervention. It's contemporary because cognitive and psychological health, as we all know, they're deeply intertwined. Disorders in these domains create major health, economic, and societal challenges. According to the WHO, there are estimates of hundreds of millions of individuals that experience cognitive and mental health difficulties and require rehabilitation each year. Now, the rise of long COVID has added new layers of complexity, contributing to cognitive and mental health symptoms in many individuals. And this leads to our European Project BRAIN, the Brain Research and Innovative Network for COVID-19, which was funded by the EU and the UK RI. Beyond its training activities, BRAIN aims to characterize and treat the cognitive consequences of long COVID. Our broader ambition is to develop accurate assessments and precision-based interventions that support individualized care. In our specialized workshops tomorrow, we will dive deeper into these topics. Now, what is the impact of the Center on Cyprus? We proudly collaborate with non-governmental organizations, advocacy groups, and patient organizations. Through our research, we have offered free assessments to thousands of individuals across the lifespan. Our participants range in age from 5 years to 96 years old, making CAN one of the few centers in Cyprus with true cradle-to-longevity neuroscience program. Cypriots benefit directly by participating in our clinical trials, helping us fulfill important gaps in health and educational services. So here's some of our achievements. Over 36 projects totaling over 23 million euros, over 1,000 publications, and over 600 conference presentations, training of about 25 postdoctoral researchers, graduating about 40 PhD and 55 master students. Anybody here graduate from our PhD and master's programs? Yeah. And providing research training to many undergraduate students. Our alumni now contribute to academia, research institutes, and the private and public sectors locally and internationally. Now, over the past 15 years, CAN has generated knowledge and technologies that can improve lives. But important questions still remain. How do we translate new knowledge into everyday practice? What are the barriers and facilitators of knowledge transfer? And in the era of precision therapies, are we there yet? We hope this year's conference can help us answer some of the above questions. We're honored to welcome our BRAIN partners from King's College, Ghent University, and Maastricht University, who are participating in today's conference and tomorrow's workshops. My heartfelt thanks go to the CAN Faculty Board, the Neurocognitive Research Lab team for their pivotal role in organizing the conference and the workshops, and the Abstract Evaluation Committee. I'd like to extend a deep appreciation to Mrs. Sevasti Unari for her exceptional organization and dedication in organizing the conference. Ms. Fotini Panagi for serving for one more year as the Master of Ceremonies and promoting today's events. I'd like to thank Emilios for helping us with all the technical things today. You're very important. And to our many volunteers who support us during a very, very busy period. So thank you to the many volunteers. So in closing, my sincere thanks to the Vice Rector, to the Dean, to the Chair of the Department of Psychology, and of course, a very warm welcome to all of you. Thank you for honoring us with your presence today, and I wish you a very stimulating, inspiring, and enjoyable conference. Thank you.
We thank Professor Constantinidou for her address. Before proceeding with our first keynote speaker invited presentation, I would like to share a few housekeeping notes. Students requesting course credit must check in and check out using the system outside the room. All attendees will receive a certificate of attendance after the conference upon completion of the evaluation questionnaire. Please keep your questions for the end of each talk during the designated questions and answers time. Most importantly, we kindly ask all speakers to adhere to the time limits indicated in the program. I would like now to invite Professor Fofi Constantinidou, Director of the Center for Applied Neuroscience, to introduce our first keynote speaker and moderate the presentation. Professor Constantinidou, the floor is yours.
So we're very excited today to have our keynote presentation by Professor Fernando Zelaya. And I will say a few words about our keynote speaker. The title of his presentation is Cerebral Physiological MRI, Applications to the Study of Brain Function in Health and Disease. And Professor Zelaya brings with him unique experiences from the industry and from academia. So after obtaining a degree in applied physics and electronics and a PhD in physics, Professor Zelaya worked in the oil industry, developing magnetic resonance methods to characterize properties of porous materials, porous media. In the early 90s, he moved to Australia, where he implemented pioneering applications of magnetic resonance spectroscopy in brain and spinal cord, and diffusion-weighted MRI in living systems. He then joined the Institute of Psychiatry, Psychology, and Neuroscience at King's College, London, where he has established advanced methods for functional MRI with special emphasis on quantitative imaging of regional cerebral blood flow and oxygen metabolism. These methods have been applied to investigations in various patient cohorts and in studies of pharmacological action in healthy control and patients. He's currently working with a small team to develop techniques for imaging blood barrier. So, Professor Zelaya, we're very happy to have you. Please welcome. I'm actually going to sit down and give you cues.
No,
yeah, when you have like 5 minutes.
Tell me when. I'm running out of time. icon. Oh, I see. Thank you so much. I'll try and stick to time. I'd like to start by thanking Professor Constantinidou and the rest of the organizing committee of this meeting for inviting me to give this presentation on the application of magnetic resonance imaging to the study of cerebral physiology in humans. I might run out of time, so I want to make sure I take a little bit of time at the beginning to congratulate you all on the amazing work that you've done over the years. It's really impressive that you've been able to have this conference for 15 years consecutively. We're very lucky in London, and sometimes we don't realize it, that we have access to resources that are not available in other countries. So I always admire the efforts of teams like yours that can do a great deal with sometimes less resources than we have. So congratulations, and I hope we can maintain our collaboration over time. So what I'll try to do over the next 40 minutes or so is to say a few things about why I think it's advantageous to derive quantitative non-invasive measures of cerebral physiology in both health and disease, and give a couple of examples of how these methods fit in the field of integrative neuroscience and psychology, which is the main thrust of this meeting. So I'd like to start always by touching base with something that all of us probably know very well, and focus on the role of functional neuroimaging in general in cognitive neuroscience. And I'll do that by beginning by stealing a phrase from Marcus Reichley in one of his seminal papers in the early 2000s, when he said that the role of functional neuroimaging is to contribute to understand how brain function supports mental activity. And that's really what drives us. And in this perspective, studying physiology is very important to see how we achieve that in the brain. It's also fair to say that apart from EEG, functional MRI, underpinned by the blood oxygen level dependent contrast mechanism that we exploited, has arguably been the most relevant technical development for cognitive neuroscience over the last 20 years or so. It's generally accepted that when there is an increase in cellular activity in the brain, there is a, let me put this pointer options here. There's an increase in oxygen availability to the neurons given by an increase in blood flow, but paradoxically, that increase in oxygen availability is far bigger than the change in oxygen utilization that the neurons execute. Therefore, this gives rise to a change in the magnetic properties of venous blood, and that is where the bold signal comes from. And although this effect is small, we've been able to reliably measure it for several years and apply it to the study of many interesting cognitive functions in the brain. A lot of my colleagues that work in this field usually begin by pointing out deficiencies of bold fMRI, but I think it's worth stressing how great this technique really is, right? In spite of some features that I'm going to mention in a minute. And I love showing this example. This is a study, 20 years old now at least, from Brian Knudson and his collaborators at the NIH, who studied anticipation of monetary reward. And it's a fantastic experiment that we still do in the department a lot. This is the monetary incentive delay task, where you see a number of cues that denote the amount of money you're going to win or you're going to lose if you respond correctly to a target. The reason I like to show it is because if you do this properly, this is presented as an event-related paradigm in the scanner. You see exquisitely distinct activation in the nucleus accumbens in the ventral striatum, which is the seat of reward processing in the brain, right? But not only that, if you analyze this and you execute this properly, you also see that the changing signal is not only located in the place where you should find it, but it's also directly proportional to the amount of reward, whether it's negative or positive, across all the trials. And this is information that you probably could not extract with anything else, right? Because the signal to noise is good enough to do that, and because this is accessing subcortical activation that you probably couldn't see with any other method in a completely non-invasive manner. So we shouldn't disregard the advantages that BOLD has given us. But the question for us, at least this morning, is what drives these signals,
right? And as I said before, the current consensus is that it's not driven by an increase in blood flow or by an increase in oxygen consumption. It's driven by the fact that these two things are not matched, right? The availability of oxygen is paradoxically higher than the utilization of oxygen, and this leads to a change in the magnetic properties of the signal. So it's important to remember that it doesn't quantitatively capture any physiological processes in the brain. Now, this becomes important when you compare bold fMRI in a cohort of healthy individuals versus a cohort of patients, for example, because you don't know where the differences are coming from. But most importantly, if you do this after administration of a psychoactive drug, because it can modify various physiological parameters and you don't have a handle of that, on that. But there is another reason why these methods are becoming, the application of imaging to study physiology is becoming more important. This is because in the last few years, there's been a growing amount of evidence accumulating that major aberrations in cerebral bioenergetics exist in neuropsychiatric disorders. So targeting bioenergetics may be an efficacious pathway to not only studying but treating these conditions too. Postmortem studies are showing in patients and patient-derived neuronal cells are showing substantial evidence of these modifications in bioenergetics in major depressive disorder, in bipolar disorder, and in schizophrenia. Differences in markers of mitochondrial function and redox biology have also led to strengthening this evidence. But most importantly, some treatments can also contribute to cardiovascular disease and dysglycemia, and I'll explain what that is in a second, in patients with mental illness. So this field of metabolic neuropsychiatry is growing. And I encourage you to read this paper. I put a couple of, I left a slide at the end with some recommended reading. This interesting review paper by Andreazzi, which touches on all of these points in more detail. Most of you know, all of you know, that the brain obtains energy by metabolizing glucose, and this can be done in two ways. The object of this is to synthesize adenosine triphosphate, ATP, and the neurons can do this with oxidative phosphorylation, in other words, using oxygen. Glucose enters the carboxylic TCA cycle and produces 36 molecules of ATP, and this is relatively slow, that's why I put it in inverted commas, compared with burning the glucose without using oxygen, which I like to call anaerobic glycolysis because it makes it very clear that oxygen is not part of this process. It only produces 2 molecules of ATP, but it's 100 times quicker. However, it leads to the accumulation of lactate, right? Which is undesirable to have in neuronal tissue. So the brain derives most of its energy from oxidative glycolysis, avoids the accumulation of lactate. But what people are beginning to observe in certain mental health conditions is a preference for turning pyruvate into lactate rather than oxidative phosphorylation, even in the presence of sufficient oxygen. So this is certainly worth investigating. Although anaerobic glycolysis exists in the brain and it supports ionic homeostasis, rapid energy production, and reduction of oxidative stress, the fact is that if it's uncontrolled, it resembles the metabolic characteristics of cancer cells, resulting in an unbalanced synthesis of ATP. So this is growing in importance, and given the high energy demands of the brain and the need to maintain functional connectivity, a shift to anaerobic glycolysis may worsen some neuropsychiatric conditions. So this is part of the reason we're giving attention to this field, at least in our institution and in other places. It can also contribute to neuroinflammatory processes in the brain. There is also the issue of redox biology, which concerns the complex reactions needed to maintain balance between oxidants and antioxidants, and this can also be compromised in psychiatric disorders and may be accentuated by a shift to anaerobic glycolysis. Of course, some types of neurons are more susceptible than others. So given the importance of cerebral physiology and the need to obtain quantitative information on it, what is a useful physiological marker to investigate? So, part of the answer lies in the process of neurovascular coupling, which you will also know probably a lot better than me, which is the process by which cerebral blood flow at the cellular level is regulated by neuronal activity directly. So glial cells release vasoactive substances like potassium ions, adenosine, and most crucially, nitric oxide. And again, from an old paper, this picture from Mark Esposito's paper shows this very nicely and describes here several stages by which vasoactive substances are produced to invade the smooth muscle of arterioles and dilate their diameter in order to provide more blood to the neurons. Nitric oxide is key in this process, of course. Glutamate binds to receptors in the end feet of astrocytes, which also releases vasodilating substances such as arachidonic acids. Astrocytes also participate in the reuptake of potassium ions, and smooth arterial muscle is directly innervated by axon collaterals that also release nitric oxide. So, regional cerebral blood flow or perfusion is a very sensitive signature of resting neuronal activity because of neurovascular coupling, and that's why we're giving a lot of attention to it. So I'll be very lucky to work with David Lithgow, Owen O'Daly, Virginia Mato at the Institute, and we have employed methods to measure CBF directly, and I'll show you a little bit of what we've done. So to establish the terminology more clearly, when we talk about cerebral blood flow or regional cerebral blood flow or tissue perfusion, we mean the nutritive delivery of blood to the capillary bed. And so it's measured in volume of blood per unit time, per unit mass of brain tissue. And the important thing to highlight is that this is irrespective of direction. We're just interested in the net amount of blood that is moving per unit time in a volume of tissue. So we have employed the technique of arterial spin labeling, with which we've had a lot of development over the last 15 years or so. And I'll tell you briefly how this works. So let's say we want to examine the blood flow in a volume of the brain, a region of the brain shown here in the coronal orientation. We select a region in the arterial input, and in this region, we apply a radio frequency pulse that we call the tag or the label. And this pulse is designed such as to invert the magnetization of that region. So we invert the signal all over the area. This, of course, has static spins, the one in the static tissue, and moving spins that are in the vascular network. If we wait a period of about 1.5 to 2 seconds, the moving spins that were in the blood, they invade the brain, and they enter the brain with their negative signal with respect to the rest, and we collect our signals as quickly as possible. Right? Then we do this again, and we need a pair of these images. We do exactly the same thing. For reasons I won't go into, we apply another radio frequency pulse, but this one does not invert the signal, right? So this time the vascular spins come into the tissue, but they're indistinguishable from the rest because their magnetization is positive. We repeat the same process. We collect our images as fast as we can. And then what we have, of course, is that in the control images, the spins from the vascular input are independent of the static ones. And in the labeled image, they're inverted. So if we subtract the two images, in every voxel of that image, we will have a difference. And that difference will be directly proportional to the amount of blood that has gone into that part of the brain. So using a model that I won't show, we can convert that difference into a map of regional cerebral blood flow in real physiological units. And this is an example of the data that we collect. And as you know, in your scanner, you have the ability to do this too. This is a cerebral blood flow map from a healthy volunteer acquired in about 6 minutes. These are some chosen sizes, and we can get this with a resolution of 2 by 2 by 3 millimeters approximately. So I'm going to show you a couple of examples of how we've employed this technique. We've been very lucky to have had the chance to do this in just under 50 studies now in the Institute. But I've chosen 2 that I think are relevant to this meeting. And this is the scary bit for me, because I'm going to talk about things that you know far better than I do. But these examples, I think, will be of more interest to you. So the first one is an investigation conducted by Leith Alexander and Basileia Koutola at the department. And this study was to examine the neural correlates of negative autobiographical memory and rumination. This is a study that I found very interesting because autobiographical memory, as you know, is the recall of personally relevant events. We all do this, And in the vast majority of cases, we remember these events, but we basically have an adaptation. If it is a negative memory, we learn from it, and this guides our future behavior. But in other subjects, it leads to what's known as rumination. So you have maladaptive behavior that comes from the repetitive, negatively biased recall of the event. So it's important, and Leith was very kind to prepare these slides with me, because it's commonly seen in depressive disorder. It's also a transdiagnostic vulnerability factor. And finally, its neurobiology is poorly understood. So there have been, of course, neuroimaging applications to study rumination, both with EPI, which is what I'm showing here. And this has identified some key areas of the brain in the subgenial of the anterior cingulate, in parahippocampal amygdala as well, parahippocampus and amygdala, and in the posterior cingulate cortex. But what they wanted to do in this experiment is to have a few methodological considerations for a number of reasons. First of all, the idea was to use personalized rather than generic stimuli to increase the ecological validity of the paradigm, to study both short-term and medium-term activation changes that occur from the presentation of negative memories. So for the short-term activation, they used bold EPI, bold fMRI, like a lot of you have done in the past. And for the medium-term activation, they used arterial spin labeling to look at blood flow changes, to use an explicit baseline, which in this instance was counting as a control condition, and to also compare with a positive emotion as a comparator in the scanner. So this repeats what I told you before, but the key issue is that they also wanted to determine if these differences in activation during negative and positive recall correlated with behavioral measures. And for some reason that didn't show at the bottom. Okay, so they recruited 21 individuals, 21 healthy volunteers with no psychiatric history at all. They were carefully screened to make sure that was the case. But they were all also screened with specific tests to establish that they were high ruminators. So they have a repetitive rumination score to measure this, extracted from this paper. They also had screening with a clinician, and they had a familiarization session in the mock scanner, and a semi-structured online interview to describe their personal negative and positive memories, with which images were made to present in the scanner. And this is the structure of the study. So they collected the normal structural images. They did a baseline observation of blood flow using arterial spin labeling with neutral images. They then presented statements relative to their memory using API. Then they presented neutral images or positive images and measured blood flow. They repeated the API using statements that relate to a negative memory, and then similarly with the ASL. So this is the structure of the API part of the study. So they had sentences to recall the memory. In this instance, the memory is associated with baby of the subject. And this was repeated several times in the scanner, and then they were asked to answer how they felt about the recall. They had a comparator task, which is the counting, and then they had a control image, and they collected API data throughout the whole examination. With spin labeling was a little bit different because the acquisition of the blood flow map takes about 6 minutes, so it was acquired whilst presenting images relevant to their own personal memory. And this gave them a map of blood flow similar to the one that I showed you before. The order of presentation of the paradigm was, of the block was alternated across subjects, and the ruminations responses were high. So the first thing they did was to ascertain if the intensity of the emotional ratings for the positive and negative images was similar, and that unfortunately was the case. Otherwise, you would not be able to tell if any differences observed in the activation were not simply driven by the intensity of the emotional presentation. So the spin labeling results are very interesting. This is the F contrast from the analysis, and it very nicely replicated some of the results that I showed you before. revealing increases in blood flow in left dorsolateral prefrontal cortex, in insula, in bilateral visual cortex, very interestingly in the posterior cingulate cortex, and in the left superior temporal gyrus. Very similar to what had been observed, of course, with BOLD. The specific post-hoc t-tests show between the positive and negative memories show specific areas in the left DLPFC as before, in the left insula, and in the left amygdala. So this is very nice to see because all these areas are consistent with what they would have expected. The EPI data was very similar too, also showing activation in the left DLPFC, in bilateral and mid-insula, in left posterior insula, and here in the anterior cingulate cortex. So with the blood flow data, they did not see any correlation with the repetitive rumination scores, but they did with the posterior insula from the BOLD data. So in the interest of time, I'm going to move on to the next study. This is a summary, basically, of what I showed you already. The rumination scores correlate with activation during negative recalls. But the interesting thing, for me at least, was that the use of stereoscopic labeling, the measurement of blood flow, highlighted limbic regions activated when recalling negative memories that show nice overlap with the previous literature, and they were consistent, as I said, with what was expected from the paradigm. The next study that I'm going to show you is was organized from the Eating Disorders Unit at the Institute, and it was led by my colleague Owen O'Daly, who has contributed enormously, and many of you met him, some of you met him back in March when you came over for the conference. The paper was written by Ethan Dalton, and from their side, it was led by Ian Campbell and Ulrike Smith. And this is an investigation of the effects of repetitive TMS to measure, to apply it to subjects with severe enduring anorexia nervosa. And they collected ASL data before and after the application of the therapy. So they recruited 34 patients with severe anorexia nervosa after assessment with the DSM-5 criterion, who had had the condition for at least 3 years, but they needed to have a BMI bigger than 14. And they also recruited 30 controls with relatively normal BMI of 20 to 25. They had a baseline scan during which they collected blood flow data and structural images. And then they were randomly allocated to either a sham TMS treatment or a real repetitive TMS consisting of bursts of 5 seconds to apply 20 times with an interval of 55 seconds. 20 sessions of this at a frequency of 10 hertz applied at 110% above the stimulation threshold. The sham consisted of stimulation below this threshold. And the RTMS was applied on the left dorsolateral prefrontal cortex because it is involved in emotion, in rumination, as you saw before, in regulation and self-control, and the application of TMS in this region has been successful in a number of psychiatric conditions. So 4 weeks later, they had another scan. Unfortunately, the dropout rate was high in this cohort. They only got back 13, all of the subjects that had real TMS, and only 10 from the sham treatment. So the numbers are relatively low. But then they had a follow-up also at 4 months and 18 months, during which there was no imaging data collected, but only clinical data, BMI, anxiety, and depression scores. So they wanted to do a whole brain exploratory analysis of the blood flow maps, but they also had a priori regions of interest in the amygdala, insula, and anterior cingulate cortex. So the results are very interesting. They didn't see any difference, actually, in baseline CBF, or in the, either in the whole brain analysis or in the regions of interest, but they saw a really interesting interaction between time and treatment, right? Which in the anterior cingulate cortex was not significant, but in the amygdala, it showed a very interesting decrease in blood flow in the real treatment compared with the sham. So this is very interesting, and this is where I think the story gets even more interesting. So 4 weeks after randomization, the change in blood flow in that region, in bilateral amygdala, correlated with the change in anxiety scores, but it was only just above the significant threshold. But interestingly, the change in blood flow also in the amygdala predicted the change in BMI in some of the subjects that gained weight. So you see the bigger the reduction in blood flow in the amygdala after the treatment, the bigger the weight gain that they experience on this side of the graph. So it's quite an interesting observation, and I know that some of that work is also done here, so you may find this interesting. The baseline value of amygdala also correlated with the anxiety score, change in anxiety score after a month. So, in conclusion, unlike other studies, this investigation did not show evidence of altered blood flow in the DLPFC, where the rTMS is being applied, which is quite interesting. But this region has indirect projections to the amygdala, of course, which may explain the results that they've observed. Subjects, as you saw, with greater reduction in amygdala CBF showed the greatest sustained weight gain at a follow-up of 18 months. Hyperactivation in the amygdala region, as you know better than me, is associated with fearful emotional processing of body images. So it's possible that the reduction may be associated with an increased ability to tolerate uncomfortable physical and emotional sensations related to the body. Now, I want to move on to, how am I doing for time? Oh, no. Okay. Okay. Well, I'll try and go quickly. We've also been working over the last 4 or 5 years or so with another technical development to examine metabolism directly. So arguably, the two most important things in terms of bioenergetics are glucose consumption and oxygen consumption. And glucose metabolism has proved very elusive for magnetic resonance imaging to do in a non-invasive manner. But oxygen is a different story. So, you know that the brain is a homeostatic organ, and from fixed principle, the rate of utilization of oxygen in micromoles of oxygen per unit mass of tissue per minute is given by this simple formula here. It's just the product, basically, of the availability of oxygen, which is what's coming in from the blood flow, times the content of arterial oxygen, multiplied by how much is being extracted by the tissue, which is the oxygen extraction fraction. So this is dimensionless, and it has a value of about 0.2 to 0.45 in healthy controls. So the brain basically balances, this is slightly moved out, balances blood flow and oxygen extraction in order to maintain oxygen consumption constant. Right? So in the face of reductions of CBF, for example, there will be a higher value of oxygen extraction in order to maintain metabolism constant. And the interesting thing in COVID is the fact that in COVID, for the first time, we encountered in a big number of subjects a condition in which the arterial content was also compromised. So I'll show some example of that later on. So can you obtain this with magnetic resonance? So the arterial content of oxygen you can obtain from just a test of hematocrit or hemoglobin from the blood. The blood flow is, of course, available from spin labeling, as I showed you before. So is the OEF determinable by magnetic resonance? And the OEF is just the difference in the oxygen saturation between the arterial and the venous side. Right? It's just whatever is being extracted by the neurons doesn't go back into the venous return, and it gives you a finite difference. Or you can define it as the ratio of deoxygenated hemoglobin over total hemoglobin. Now, you know from ball fMRI that MRI is sensitive to the magnetic properties of hemoglobin. So there is certainly a possibility to measure this. I'll show you how we do it, but I'm going to do it with pictures. And I'll try not to put anybody out, make anybody leave at this point from showing too much physics. This is a lovely T2-weighted image. It's the axial, transverse axial size through the brain. And it's lovely for several reasons, right? The contrast is fantastic between cerebrospinal fluid, white matter, deep white matter, and gray matter. But it's also lovely because there's no distortion. Right? There's signal everywhere, and you see a great deal of anatomical detail everywhere. It has a great resolution. And what it has, however, it has severe macroscopic and microscopic inhomogeneities. Failure, inhomogeneity, we mean spatial uniformity of the field. So, but it's acquired with something called a spin echo, and the spin echo is very good at reversing that. So this is the degraded spin echo on the left-hand side, right? And you see that if we collect it with this other thing called gradient echo, then we reveal these macroscopic field inhomogeneities. And this is very useful clinically, right? Because the image on the right-hand side is what you would use. For example, if you want to investigate cerebrovenous malformations or microbleeds in the brain, you want to be sensitive to those distortions, right? And so do we, if you want to study oxygen metabolism, but we want to only be sensitive to the microscopic field inhomogeneities caused by deoxygenated hemoglobin. So fortunately, there is a way to separate these two effects, and we do it with this thing. I apologize for this slide. It's not animated. There's a lot of information on it, but I'll take you through it slowly. So normally we generate the signal, which is shown here with a radio frequency pulse. We call a 90-degree radio frequency pulse, and the signal decays. That's how we normally acquire a gradient echo that I showed before. But if you put another pulse, 180-degree radio frequency pulse, you can regenerate the signal to almost the same amplitude that you had, and this is the spin echo with which normally we acquire the nice image that I showed you before. But somebody had this great idea of moving the 180-degree pulse towards the 90-degree and keeping the acquisition window in the same place. If you keep the acquisition window constant, you get a very funny-looking echo, right? And this echo is very powerful because the more you shift the 180-degree pulse, you see the images get darker and darker. But if you plot the intensity of that signal versus the value of the shift, it turns out that the slope of the logarithm of the signal versus the shift is linear in this region. And it's directly proportional to the oxygen extraction fraction times the deoxygenated blood volume. So I didn't invent this, I wish I had. But it can very powerfully allow us to separate the macroscopic from the microscopic component, which is what we're after. So I'm very lucky to work with David Lithgow, who has developed a technique called gradient shimming, with which you can do that, right? So what we did was incorporate that into that pulse sequence that I showed you before. These are some of the publications that David has done in the field of spinal cord imaging, and I have an example here. If you acquire a normal gradient echo, you don't see a great deal below the jaw in the cervical spine. But if you understand, the lights don't do justice to the image. But it doesn't matter. You collect you can observe a great deal more tissue by adding gradient shimming to that acquisition process. So we included that into our pulse sequence. Those are the little gradients here. And these gradients are tiny. They go from minus 100 to a plus 100 microtesla per meter. And they are derived from a field map that's also acquired over the brain. And so using this, we can compute the slope of that signal change. And this is very simply given by this product. And this factor here, delta omega, is just a change in frequency caused by the oxygenated hemoglobin. That is fortunately given by this a formula that looks a bit horrible, but everything in that formula is either coming from textbook or from the experiment itself. And the only unknown is the concentration of the oxygenated hemoglobin, which we can therefore infer and divide by the total hemoglobin to get our map of oxygen utilization. This is some of the data that we've acquired in healthy volunteers. Unfortunately, it takes a little longer to acquire, 8 minutes, right? But we can do it with relatively good spatial resolution. And we have been able to examine the reproducibility of the technique across several regions in the brain. It's not very good, at least in 3 areas, genial of the ACC. Unfortunately, in some parts of the ACC, here is quite low, but everywhere else is nicely well above 60%. So I'll show you very quickly this. I'm sorry. I'll quickly show you the results of this study conducted by Daniel Martins, and he used this method in people who had COVID. So the 40 subjects were recruited. They all had mild COVID infection. 20 of those had persistent fatigue, chronic fatigue, and 20 were recovered. Okay, they were all scanned with structural imaging, arterial spin labeling to determine blood flow, and the OEF sequence to determine the oxygen extraction fraction maps and compute the oxygen metabolism. So the regions of interest a priori were the hippocampus, anterior insula, anterior cingulate, amygdala, and striatum. And they also executed the cognitron battery, or parts of the cognitron battery, which I think you have now available here too. And these are I'm not going to read them all, especially because I have no time. These are the paradigms that they did. Okay, so in the blood sample analysis, they observed higher creatinine and gamma GT levels in those subjects who had persistent fatigue. The platelet count also tended to be higher, but all the other metabolic markers were unaffected. Interestingly, the TNF-alpha measurement suggested evidence of elevation in the persistent chronic fatigue group. In the cognitron tasks, they showed significantly poorer performance on delayed object memory and trend level impairments in this abstract reasoning task, but all the other domains didn't show any difference. Global perfusion was preserved in both cohorts, but there were substantive regional differences in blood flow in these areas, increases and decreases of blood flow, increases in the anterior and mid-ACC, bilateral insula, the superior temporal gyrus, caudate, putamen, and thalamus, and significant reductions in the left posterior cingulate cortex, occipital cerebellum, and parahippocampal gyrus, in the absence of any gray matter volume changes, which is interesting, because of course this could be driven simply by a loss of gray matter, but that was not the case. In terms of oxygen metabolism, most of it was preserved except in these regions here, in the ACC and in the hippocampus, and Daniels also showed that higher hippocampal metabolism positively correlated with cognitive performance, suggesting some compensatory adaptation to carry out the task. The lower CMR2 in the ACC correlated with depressive symptoms, with reduced motivation and elevated TNF. And this is being prepared for publication at the moment. It's under review, and it will hopefully soon be available. So I'm going to skip this part due to the lack of time. I wanted to show some of the work that my colleague James Paul is doing in terms of obtaining quantitative markers of blood-brain barrier function and glymphatic activity in the brain, but we can either revisit this in the panel discussion or address it in the workshop tomorrow. But one of your aims was to make this contemporary, and I think this is probably one of the most contemporary things going on in terms of physiology and neurofluid investigation in the brain now. So I just want to thank my colleagues who have worked with me, and Steve, who had faith in this, enough faith in this kind of methodology to support us throughout the years to get some of the work done. Thanks again for inviting me and for your attention. Do you want me to?
Yeah, thank you so much. Yeah, I think we have time to take one or two questions. So if we have one or two questions from the audience. Caldia, yeah.
Thank you. Thank you, Fernando, for this very interesting presentation of yours, and thank you for being here today. My question is, how can the physiological MRI tell the difference between brain activity and changes in blood flow in people with long COVID, for example? How can tell that a signal comes from a brain activity or changes in blood flow?
You mean if you were to do a cognitive challenge and do all the MRI?
Yes, during a cognitive task.
Yeah. So you would have to then do an ROI interrogation of the value of blood flow and OEF in the activated region and determine if there is a physiological change in that area. Normally, the signal increases if blood flow is reduced, right? So you could explain the signal change, whether if it was higher, by an observation of reduced CBF in that region, right? Or you could observe a lower OEF in that area, right? Which would mean that there's less oxygen utilization, which also would account for the increase in the Bolt signal. But the answer is you would have to do these two investigations. Three, actually. You would do your cognitive challenge with Bolt MRI, and then you'd have to measure CBF with ASL, and then you'd have to also have time to measure OEF in that area. But it's important to and this is a very good question, because it's important to highlight that the two investigations of blood flow and OEF in that case, they would be in the resting state, right? Okay. So it would be to answer whether any resting physiological differences account for the observed differences in activation. Ideally, you would do the task with arterial spring labeling, right? But one of the things I did not mention is that labeling blood flow, waiting for the labeled blood to go into the brain, and then acquiring new data with modern methods takes about anything between 3 and 5 seconds. So that temporal resolution is too low to do any cognitive tasks with. So if it was quicker, then you would directly measure the blood flow in the presence of the cognitive challenge. But we don't have the ability to do that.
Thank you. Thank you.
Any other questions?
Thank you for the presentation, Fernando. Just an fMRI question to get your view on. There's a recent trend, I think, towards using comparative tasks as a baseline and not just a resting state baseline. And I just wondered what you thought about that.
Oh, you mean a resting state fMRI in addition to?
Yeah, using a comparative task, something that is very similar to the experimental task, just to get the comparison, not just the resting state.
I'm not sure I understand your question. So say you have a semantic task with bold fMRI. So do you mean to do another challenge?
Yeah, which you will compare to the experimental, because it is very different from the resting state. So I guess if there's any more value to adding the comparative task rather than just getting the resting state.
Yes, but yes, of course. But I mean, your question touches on a really interesting thing. If for any reason you don't have enough time or you don't have a sequence in your scanner to measure blood flow and oxygen metabolism or whatever else, then it is really valuable to do a simple sensory task, because any change, let's say especially if you have 2 cohorts, right? You would want to establish that neurovascular coupling is not impaired, right? That changes in neurovascular coupling are driving the task. So if neurovascular coupling is not different across, then the level of activation of the sensory task should be the same. But if you don't observe that, then that gives you an idea that there is something else driving the changes that you have. I don't know if that answers your question.
It does, yeah. Thank you.
Okay.
Thank you so much.
Thank you. I'm sorry I went over time. Yes.
Thank you so much. Now it
Thank you.
works. Yeah. All right. Congratulations. Okay, for this, thank you again, Fernando, Professor Zelaya. Also, as part of the BRAIN project, one of the tasks was to harmonize imaging protocols used by our team in Cyprus to the UK biobank imaging protocols. And so we did that with the participation of Steve and the whole group and Fernando. Also, our team has received training. We here in Cyprus, and also we were in the UK last winter, where Fernando led a team of researchers that trained our team on the different methodologies. So this is part of the capacity building of the project. So thank you again. Okay, so for the next part of the conference, we're going to present data from the BRAIN project. But before we do that, as part of the project, our consortium included here in Cyprus colleagues from the UCY Medical School. And one of our colleagues is Dr. George Nikolopoulos, who is an epidemiologist, and we thought that it would be a great opportunity to hear from him about the pandemic and the current trends. Some of you may remember that Dr. Nikolopoulos was part of the the team that presented during the 2020 Cannes conference. We did a hybrid conference at that time where he showed us the trends of the pandemic, and we were still in the first phases of the pandemic. So, Dr. Nikolopoulos.
So good morning, everyone. Thank you, Fofi, for the introduction and this nice invitation to remember a couple of things, primarily epidemiological and public health related to COVID-19. I will try to cover some parameters that have been studied in the past, but they are relevant today, current trends, and also future challenges. And I hope this will be a nice introduction to the actual results from the BRAIN project. You probably remember that it all started in late December 2019 with some cases, serious pneumonia cases of unknown etiology in China. However, in early January 2020, the scientific community successfully responded to that threat, and the causative agent, a virus, the severe acute respiratory syndrome coronavirus 2, was discovered. And just to help you with a comparison, when HIV, the virus that causes AIDS, another pandemic, when AIDS cases actually were diagnosed in the United States in 1981, it took 2 years to discover the virus and then develop tests and so on. Now, in a matter of days, the new virus was discovered, as you can see here, its structure. And more importantly, the whole genome was sequenced, which allowed researchers to see the similarities with other coronaviruses that circulated in humans or in bats, as you can see in the phylogenetic tree right here. And as it happens always with a pandemic strain, there was a fast spread everywhere globally. And in early March 2020, the World Health Organization declared a state of global health emergency, which lasted approximately 3 years. One of the research questions early in the pandemic, and it remains until now, is the origin. As I said, the virus shows great genetic similarities with viruses that circulate in bats. So this was the origin from bats. The question is whether it was directly a transmission from bats to humans or through an intermediary host. It seems, and this is the most likely scenario. Other hypotheses cannot be ruled out, that probably this animal here, a raccoon dog, was the intermediate host. The other highly politicized question is whether this was a natural spillover or a lab leak. Both hypotheses are here. My personal opinion, and based on the evidence and the scientific opinion of well-established scientists, is that it is probably a natural spillover, because these things have happened in the past multiple times, are occurring right now, and will happen in the future, so we have to be prepared. In terms of transmission, another topic where there was a knowledge shift over time, and this is something we should know in science, that our knowledge could change over time, but we have to be clear on this issue, how we communicate messages, especially to the general public. Initially, many infections were attributed to touching inanimate contaminated objects, or that it was a direct transmission through droplets from one infectious person to another who was in proximity, but it seems that there is a more general transmission through the air, which includes inhaling of aerosols. So there are particles suspended in the air, which could be inhaled not only in short distance, but it seems also in longer distances, and especially indoors. Therefore, measures that were recommended, social distancing, masks, and good indoor ventilation had a good scientific rationale. In terms of mortality, people died. This was a serious pandemic. These are the official numbers in so far, so confirmed deaths more than 7 million. We have, however, to be clear here that there was an age-related mortality pattern, which means that older people were more likely to experience serious COVID-19. And because there were some arguments here, again, how many people died, there is a very good indicator that was used during the COVID-19 pandemic, epidemiological indicator, which is the excess mortality. So we compare what is expected to happen based on historical patterns and based on what is observed, the difference. This is called excess mortality, and there have been some very nice studies published in high-quality journals which showed, yes, there was excess mortality due to COVID-19, not only in European settings, but also in lower and lower middle-income countries. In terms of the effectiveness of the measures that were implemented, again, a lot of discussions and debates, which make sense. I have to say here that it is very difficult to conduct randomized clinical trials, especially during emergencies, to evaluate effectiveness. And another problem is that you cannot study these measures in isolation, because when there are emergencies, many measures are implemented at the same time. However, there is good observational evidence and from mathematical modeling, and this is a nice study that has summarized more than 300 research papers on this issue, which has shown clearly that all these measures, including stay-at-home orders, reduced transmission and consequently saved lives. But there were also studies which compared settings with more stringent measures to settings with less stringent measures, and they found association of these measures like mask use requirements with hard outcomes like death. I acknowledge that this debate will not stop, it will continue. The evidence is that they were effective in stopping spread of COVID-19 and saving lives. On the other hand, however, there are impacts perhaps in other diseases like mental health issues or disruptions of economic activity, of the educational process. As societies, we have to discuss seriously what kind of balance we want, how many lives we want to save now against some consequences in the future. Vaccines, another hot topic. Unfortunately, although the evidence has been clear, not only for COVID-19, but for other infectious diseases, they saved lives. These are two nice papers. Perhaps the numbers are not very clear. They were published in 2024, I think, and 2025, and both showed that more than 1.5 to 2 million lives, primarily among the elderly, were saved. In terms of safety, because this is another issue, there have been thousands of studies. There are routine systems that monitor and evaluate the safety of vaccines, and whatever adverse event was, it was detected, like the rare clotting and bleeding condition associated with adenovirus vaccines, which were removed from circulation, or some rare events of pericarditis or myocarditis associated with mRNA vaccines. But again, these were very rare events. They have been captured. And in addition, and this is a very recent study, November 2025, which showed that in children, the incidence of the same event when people acquire naturally the infection is even higher than the incidence of this adverse event when people are vaccinated. So the message is vaccines are effective and safe. In terms of the current situation, we have been transitioning to an endemic circulation of COVID-19. The WHO continues to receive confirmed cases of COVID-19, but perhaps the best way to monitor COVID-19 is through an integrated surveillance, sentinel surveillance, that covers not only COVID-19, but also influenza and RSV. And fortunately enough, at least in Europe, these systems have been working, and you can see the positivity rates in tests. Now, in mid-November, it seems that more tests are positive for influenza. And the influenza curve is to the left. It started, it's increased because it has a seasonal pattern, and the green curve is the last wave of COVID-19 in Europe, which seems to be receding because COVID-19 has not a traditional seasonal pattern yet. It could occur in any month. Therefore, we still need vaccines, especially the elderly and people with comorbidities, and their safety and effectiveness are associated every year. And these are very recent publications which showed that there is a protection. Keep in mind, this is another message. We don't have 100% protection. This is hard to achieve. However, even 30, 40, 50% protection, especially against serious disease, is important for people who need it. And of course, something that you have been doing some great work, there is also the post-COVID sequelae, which is important, the post-COVID-19 condition. It is real. It has been studied extensively. There are wonderful publications which have showed that it may affect multiple organs, including the brain. And although some people are more likely to experience it, all of us, even after a mild infection, could have it. So it is important to continue research. And in terms of the future, COVID-19 virus will stay with us probably forever. Therefore, we have to monitor the infections. Post-COVID condition will happen, so we have to invest the resources, and that's what the BRAIN project has been doing. We should continue to offer vaccination to those who need, and something else, we also need better ventilation indoors. And finally, keep in mind that it's not only a COVID-19. We may experience in our lifetime another serious event. I hope not to experience it, at least those of us who are older, but it could happen. Therefore, we have to prepare. And this is something that has started at the European level. There is a lot of investment, important to detect early, to improve our mathematical projections real time, to improve digital contact tracing, as I said, to improve infrastructure for indoor ventilation, and more importantly, to fight against the misinformation and disinformation that undermine public health and especially vaccines. Thank you, Fofi.
Thank you.
And thank you, all of you.
Thank you. We can take one question while the team is setting up the other presentation. Yes.
That was brilliant, thank you. And hopefully the Center for Disease Control in America uses that information to inform the senior politicians about what the vaccinations. Have you looked at the impact of social isolation on mental health? In the UK, we're really worried about the severity of the lockdown and the long-term impact on particularly adolescent females in terms of general psychiatric health and well-being.
To be honest, I have not read every single paper of those included in this review I mentioned of the impact of interventions. I think there are studies, or we may have studies in the future. And this is what I mentioned, that we have to evaluate the entire impact of these measures. Yes, they reduced infections, they saved lives, but certainly there were negative impacts. And we have to discuss then what we need when there are emergencies, because decisions have to be taken when something is spreading rapidly. Should we save people's lives, including the elderly, because for me it makes no difference to exclude the elderly and to protect them magically somehow and isolate them in their homes? Should we save lives now and have some negative consequences later, or we take the mid and long-term impact more seriously? So I don't remember whether some studies, including especially in the UK, because they have done some good work, but even if there are not, we have to do it, to evaluate the entire impact and then take decisions, especially now that there is no emergency, what we should do when things are hot.
Thank you.
Thank you so much.
Thank you.
Thank you again. So now we're going to move on to talk and share with you some of the findings of the BRAIN project. I will spend a few minutes also introducing the BRAIN project. And in addition to myself, there are several members of our team that are going to be presenting. Okay. All right, so this is the consortium. We have the University of Cyprus as the coordinating organization, the Center for Applied Neuroscience, and then collaborations with the UCY Medical School, the Department of Psychology, and the GIOS Research Center. Then we have our partners from Maastricht University, from the Faculty of Psychology and Neuroscience, from Ghent University, and then from King's College of London, Department of Neuroimaging, Center for Neuroimaging Sciences with Steve Williams and Fernando. So, Steve was asking about what are the effects of the pandemic on mental health. And very early in the pandemic, our team embarked on a study during the first wave of when Cyprus established very strict measures to control the pandemic. And at that time, we had the first death, and we conducted a study looking at the psychological effects of isolation and the impact of COVID in the general public. The majority of the general public, we had not had the infection at that time. And we found increased levels of anxiety and depression in our cohort, which was primarily working age cohort, and especially higher rates in younger people, like undergraduate students. Then we did a follow-up study, and this is the study that you have here, where we investigated using the same questionnaires, but also added an impact questionnaire to look at post-traumatic stress in the fall of 2020, just as there was an increase in cases of COVID in Cyprus and elsewhere. And there were starting discussions about a second lockdown, where we found even higher levels of anxiety and depression. We found PTSD symptomatology, but then we also found that those who had better coping and resilience fare better. That should not be a surprise to those of you who do research in mental health. But it provided some guidance about perhaps vulnerable groups like the younger adult group, the undergraduate students, and the elderly. And we provided recommendations to the Ministry of Health about how they can maybe do more targeted campaigns for those more vulnerable groups. It also raised questions for us regarding perhaps if certain populations will be primed to sustain long COVID because they already have higher depression and anxiety symptoms, even without having a history of the disease, just because of the pandemic. So those were some of the questions that kind of led us to a series of studies, and we participated in a number of different studies, international surveys, a European survey for student well-being, and then also other international surveys looking at sleep, the effects of sex, where we're finding some consistent findings in terms of the effects of COVID, social distancing in the general public. So the other issue that is of concern to us was the post-COVID condition, in that early in the pandemic, there were studies demonstrating neurologic and other psychiatric effects of people who had recovered from the initial infection. And it's thought that in addition to dementia, brain injuries and stroke, cancer treatment, long COVID and other infectious conditions are driving the need for cognitive rehabilitation services and also driving the cost of cognitive rehabilitation. So those were some of the findings and dynamics that led us to consider exploring and investigating the post-COVID condition as part of our twinning project. So the project addresses the specific challenges of building excellence capacity through an integrative neuroscience network that will enhance research and innovation resources in the widening country, that's Cyprus. BRAIN will generate scientific and technological and societal impact by giving a strong impetus to applied neuroscience research in Cyprus and the broader Mediterranean region, and contribute to the training of new generation of highly qualified researchers and clinical scientists. And the BRAIN project focuses on PCS, of post-COVID syndrome, of post-COVID condition. There's different names referring to long COVID. As this clinical category is relatively new, it has created unprecedented and unmet needs worldwide. However, the established methodologies that we have implemented through the project can really be transferable to other conditions that are of interest and that affect mental health and produce cognitive deficits. So there are a number of key activities that we have embarked over this period of 3 years. The project started in January of 2023, and it's continuing through June of 2026. So when we talk about the long COVID condition, there's two definitions. One definition is the WHO definition. The other definition is the CDC and the NICE definition. Our team follows the WHO definition, where it says that it's a constellation of symptoms present at 3 months after the initial onset of the COVID infection. And this is symptomatology that is persisting for a duration of time, for at least 2 months. And what are some of the thinking and emotional effects of long COVID? It includes mood and emotion changes such as depression, anxiety, and PTSD. It includes physical health issues such as pain, fatigue, headache, voice and swallowing problems. It causes problems in thinking such as brain fog, concentration, memory difficulties, difficulty with planning and executing goals, and also word finding, and also behavioral changes, including sleep problems, relationship problems, substance use, and social withdrawal. So the research considerations of the brain project is to attempt to characterize the neuropsychological phenotype of the condition. Is it predominantly an affective phenotype? Is it a cognitive phenotype or variant? Is there evidence for neurodegeneration? And the duration of PCS, and to also characterize the duration of the PCS symptoms based on clinical symptoms and possible phenotypes. We also wanted to see if there were differences as to when someone contracted the virus in terms of the variant type, like the first wave, the wild type, which was very severe, versus the Omicron variations. We also wanted to explore sex and age differences. We wanted to look at the contribution of cognitive rehabilitation in improving cognitive difficulties. We also wanted to see the feasibility of incorporating neuromodulation methodologies along with cognitive rehabilitation to improve cognition. And ultimately, the BRAIN project will lead larger studies to determine treatment efficacy and maybe in the future identify which is the most effective rehabilitation pathway based on those phenotypes. So it's pretty ambitious. But when we're talking about levels of evidence, what is the level of evidence? One, for characterizing, and two, for treating the neuropsychological deficits associated with the long COVID condition. Where are we in this pyramid, starting with more background research, individual case studies, eminence opinions, more of expert opinion. When we started investigating COVID and the COVID condition, we were at the very early stages, the bottom of the pyramid, right? How much progress have we made? And so one of our first studies was a systematic review led by Dr. Eva Betemeridu to look at the cognitive and psychological symptoms in the post-COVID condition, systematic review of structural and functional neuroimaging, neurophysiology, and intervention studies. And some of the key takeaways of that review was that there are significant cognitive issues, especially in regards to attention, executive function, and memory across 78 studies involving more than 5,900 participants. The evidence showed mixed results regarding structural and functional brain changes, including gray and white matter volume, cortical thickness, functional connectivity, electrophysiology, and cerebral blood flow. Non-invasive brain stimulation techniques have some promise, especially TMS, transcranial magnetic stimulation, and TDCS, transcranial direct current stimulation. And those potential benefits are from managing depression and cognitive deficits. So in terms of when we started the study, where was the world in terms of COVID at the time? We had our kickoff meeting around the 20th of January, 2023. There were about 665 million people confirmed cases worldwide at the time. Now, where were we last Monday? We added about 100 million cases. So the growth is not exponential, right? Is it because there is a true reduction in the infections because of immunity or vaccinations? But is there also a reduction in reporting? Probably both, right? Both things are occurring. And then in terms of Cyprus, since this was a post-COVID study conducted in Cyprus, to date we had about 700,000 infections. Remember, some of those are repeat infections, excuse me. But then in terms of looking at Cyprus, in terms of number of infections per 100,000, we are about 31,000 per 100,000. I'm sorry, the US is about 31,000 per 100,000, where Cyprus is about 85,000 per 85 per 100,000. So really, we have a lot of cases. And the question is, where are the long COVID patients in Cyprus? Where are they serviced? Where are they seen? So we'll be talking more about that. And I'm going to turn it over to Dr. Maria Constantinou to talk to us about the epidemiological or cross-sectional data from Cyprus. Hello, everyone. I'll talk about the cross-sectional study that we conducted for the Project Brain. The aim was to understand the psychosocial and cognitive health post-COVID-19 in Cyprus. So the first thing we did was we created an online platform for people to be able to participate in our study from any electronic device, their smartphone, their tablet, their laptop. We implemented the first part in RedCap, which consisted of surveys to collect demographic information, information related to COVID-19, and also to conduct a brief psychosocial assessment. And then we used Cognitron, which was developed by our partners in King's College London, in order to do a cognitive assessment with interactive tasks, which are all implemented online. Importantly, when we collected data from February 2024 until May 2025, and this is what we're presenting here. As this was a long battery, we reported the people that completed every part. So 546 people started the survey. Out of this, an 80% completed the surveys. After cleaning the data and removing duplicated records, a sample of 416 people was analyzed. The demographics are shown in the red box next. Out of these people, 90% of people have reported that they had contracted COVID at least once. And out of these people, around 74% also completed all the tasks of the cognitive assessment. In these two plots, you can see when people in our sample had contracted COVID for the first time and or for their last time. People had reported contracting COVID multiple times. You can see the distribution of how many times people had contracted COVID in our sample. And to analyze, to understand the effects of COVID-19 on in our psychosocial in psychosocial health, we looked at people grouped according to when they had contracted the COVID for the last time. Importantly, in our survey, we have asked people, how well have you recovered from COVID-19? And 30% of people said they had not recovered fully yet by the time they had took the survey. Participants in Cyprus reported experiencing a range of symptoms post-COVID, which is comparable to other studies internationally. The most common neurological symptoms that they reported were brain fog and also headaches or migraines. Other common symptoms included fatigue, general symptoms that got worse after physical or mental effort, and muscle or joint pain. In our psychosocial health investigation post-COVID, we have measured levels of anxiety, of depression, or fatigue, and of health-related quality of life. And after looking at these levels compared to when people contracted their last COVID-19 infection, we found that health-related quality of life had a statistical significant effect and significant difference between groups of people that based on when they contracted COVID, but anxiety, depression, and fatigue did not have a statistical significant difference whether people had contracted COVID soon or longer from when taking the test. For cognitive assessment, we used the interactive tasks measuring a range of cognitive functions, including memory, problem solving, semantic reasoning, attention, executive functions. Here you can see how the tasks looked. And out of this individual task, we computed a global cognitive score and used regression analysis to understand how different COVID-related factors might relate to the cognitive performance. We found that age and education, as expected, was the main predictors of cognitive performance. And also from the COVID-related parameters that we examined, the number of COVID-19 infections, which also includes zeros as for people that did not contract COVID, was also a significant predictor of cognitive performance. Other parameters that we used, such as the time since infection and an estimation of the variance when the first infection, when people had the first infection, was not a statistically significant predictor. And also the levels of anxiety, depression, fatigue, and health-related quality of life were not predictors of cognitive performance. And this was a small snapshot of our results from the cross-sectional study. And now Dr. Iulia Solomou will talk about the feasibility study that we contracted as part of BRAIN.
Hi, everyone. So the aims of this feasibility arm were twofold. First, we wanted to examine whether the integration of a neuromodulation method with a neuropsychological therapy could be implemented safely to post-COVID syndrome. And also the second aim was we wanted to generate guidance for larger clinical studies by understanding what it works best for this population. So as you can see here, the combined intervention was quite intensive. Participants completed a 5 weeks program, attending 3 sessions per week for a total of 50 sessions. Each session included 90 minutes of cognitive training and 2 blocks of TDCS stimulation separated by a short break. So participants were doing real cognitive work while also receiving stimulation, and our goal was to understand whether this type of combined protocol is feasible in long COVID patients. For the cognitive component, we used the categorization program, a well-validated intervention with strong evidence in traumatic brain injury, healthy aging, and mild cognitive impairment. The CP targets 2 fundamental areas of cognition, recognition, and categorization of everyday objects, and new category learning and concepts formation. So now, traditionally, the CP is delivered using paper and pensive materials. However, we wanted something more convenient, more engaging, and also compatible with the home-based rehabilitation. So we digitalized the entire protocol and created CatBrain, a cloud-based software version of the CP. And this allows us to deliver the task consistently, track the progress automatically, and give participants a familiar screen-based format rather than a printed materials. So this is what the platform looks like. You can see on your on the right part of the screen what the therapist screen, and on the left side you can see the participant screen. So at that point, we had a validated cognitive rehabilitation program, and we had the capability to deliver it digitally. The question becomes, what happens if we add TDCS? Can these two interventions be combined in a way that patients can tolerate, complete, and perceive as useful? And to answer that, we designed the feasibility study. You can see here we had a comprehensive pre-assessment, then allocation to the CP plus active TDCS or CP plus sham TDCS, and finally we had a comprehensive post-assessment. And here is the feasibility matrix we used to evaluate whether these combined approaches could realistically work. We focus on the domains of recruitment, adherence, and acceptability. Altogether, these measures helped us determine not only if the study could be done, but also whether it was meaningful and well received by the participants. So this slide here summarizes the recruitment process for our feasibility study. In total, we initially identified 131 potential participants. 80 of them came from the post-COVID clinic, and the rest were identified through social media and community screenings. Out of those 103 participants were assessed for eligibility, and 17 could not be reached or had duplicate records, and 51 were excluded for different reasons. The most common one was that they no longer experience post-COVID symptoms. So from the 103 eligible individuals, 28 completed the initial assessment, giving us a recruitment rate of about 27%. Now moving on to the adherence, here we wanted to see how many participants actually followed and completed the intervention as it was designed. From the 16 participants who started the 5 weeks intervention, 12 completed it, giving us an adherence rate of 75%. So the main reason for discontinuation were mild side effects or concerns related to the TDCS in one case, and the lack of time in the 2 other cases. So the overall, this level of adherence is quite encouraging for a feasibility study, especially considering the demands of the 5 weeks combined intervention. So this slide here presents the results for acceptability, which we assess using functionality questionnaire completed after the intervention. 11 participants rated their perceived improvements across different daily domains and cognitive skills, such as memory, problem solving, and etc. So the overall results were positive, with most participants rating their improvements between good and very good. And an average score ranging from 3.8 to 5 out of 6. The highest ratings were observed in the maintaining independency, social interactions, and problem solving, suggesting that participants found the program both useful and relevant to this everyday functioning. To complement the questionnaire findings, we also conducted semi-structured follow-up interviews to better understand participants' experience with the intervention. Several common themes emerged. Participants emphasized the importance of the therapeutic relationship in maintaining their motivation. One participant even said that if I didn't like it, I wouldn't have completed the intervention, highlight how crucial the rapport was for the engagement. Others described feelings of cognitive clarity and renewed sense of mental sharpness, and many also mentioned improvements in word finding during daily activities, suggesting that program had a noticeable impact on real-life functioning. So overall, this quality feedback reinforced the acceptability and perceived benefits of combined cognitive training and TDCS protocol. And now my colleague, Flora Nicolaou, will continue by discussing the preliminary neurocognitive outcomes.
So, as Julia told previously, our sample included 43 participants who completed the baseline assessment. 14 were men and 29 women. From this group, a smaller subset continued into the feasibility phase of the intervention. 5 participants received the cognitive rehabilitation combined with active TDCS, while 7 participated in the cognitive rehabilitation with non-active TDCS condition. Moving on, these are some of our preliminary results about the participants who completed the baseline assessment. Our participants range in age from 29 to 64 years, with a mean age of about 50 years, and had an average of just over 5 years of education. In terms of cognitive scores, the mean of mini-mental status examination was 28, suggesting a generally preserved global cognition. Psychologically, the GAD and the PHQ means were around 6 and 8.5, reflect a mild to moderate anxiety and depression and depressive symptoms. Before I move into the neurocognitive test findings, let me briefly describe the independent CP test that we use to evaluate the efficacy of intervention. The categorization program includes its own standardized assessment tools that directly measure the skills trained during CP. These tests are assigned to parallel the structure of the intervention itself. CP Test 1 reflects Part A, which focuses on object categorization tasks, where CP Test 2 and 3 reflect Part B, the new category learning task. These CP tests allow us to capture target changes target changes in categorization abilities beyond what neuropsychological tests can show. So these are the active ingredients of CP, the progression from concrete to abstract, the strategies to improve classification skills, working and episodic memory, cueing hierarchy to facilitate learning, intense training and stimulus redundancy, and executive functioning. We performed a pair sample t-test comparing pre- and post-training performance regardless of whether the participant received active or TDCS. Here we can mainly interested in whether the categorization program itself produced measurable gains. Here we are mainly interested about the categorization program, and for CP Test 1 we observed statistically significant improvement from pre to post intervention. From CP Test 2 we again found a significant improvement, and for CP Test 3 we also observed a higher mean score at post test, indicating a positive trend, but the difference did not reach statistical significance. This could be due to the low complexity of the task, and we have observed in other studies that some high-functioning individuals reach ceiling effects. Overall, these preliminary results suggest that participants showed meaningful gains in the categorization skills, specifically targeted in the CP training. These are the preliminary results from the subgroup who proceed with the treatment. We have 12 individuals starting the intervention. Their average age was about 50 years, similar to the larger cohort, and education was again around 5 years. Looking at the baseline cognitive and psychological scores, global cognition on the mini-mental status examination remained in normal range, while delayed recall, which administered again we administered again the HVLT, and the delayed recall was slightly reduced. Anxiety symptoms were mild on average, and depressive symptoms were somewhat elevated. This slide shows our pre- and post comparisons for participants who completed the full intervention. We wanted to examine changes across cognitive and psychological measures, and as you can see, there was a trend toward improvement in mini-mental status examination, which is just above the conventional significant threshold. This suggests a possible enhancement in overall cognitive functioning, even with this small feasibility sample. For memory, as measured by the HVLT, delayed recall was not statistically significant, as well as trail-making performance. On the emotional measures, there was a non-significant decrease in GAD scores, indicating potential reduction in anxiety symptoms, while PHQ scores remained stable. Overall, these findings show no statistically significant differences yet, but the direction of change is consistently positive, supporting the feasibility and potential benefit of the combined intervention. Also, it is important to note that emotional measures are screening tools and not test the confirmed diagnosis of the participants. Now I will present you two case studies, one individual who received cognitive rehab through CP and active TDCS, and one individual who received cognitive rehab through CP but no active TDCS. This is a case of a 60-year-old female participant who received cognitive rehabilitation, and her cognitive performance shows in the graph improved across several areas, particularly in attention, movement speed, and memory-related tasks consistent with the overall group trend towards cognitive enhancement. Cognitive rehabilitation tests, pre and post, confirm again improvement that mentioned earlier. When we look at the self-report measures, her GAD score decreased from 10 to 5, suggesting a reduction in anxiety, whereas PHQ increased from 7 to 11. This rise in depressive symptoms might reflect on greater emotional awareness or sensitivity following the intervention rather than engine deterioration, something we have seen echoed a qualitative interview. However, her SNAP score declined from minus 15 to minus 70, indicating that she perceived more pronounced cognitive and emotional difficulties after the intervention, and it seems that awareness of symptoms increased. In case 2, a 64-year-old female participant who received cognitive rehabilitation with non-active TDCS, meaning she completed the categorization program alone. Her cognitive profile shows relative stability across most domains, with small improvements in reasoning and spatial memory tasks. This supports the idea that the categorization program on its own may help maintain or slightly enhance cognitive performance even without the active stimulation. At the same time, again, CP tests pre and post show improvement as mentioned earlier. On the self-report measures, her PHQ increased slightly from 7 to 8, and her GAD raised from 7 to 11, suggesting heightened emotional awareness or stress sensitivity toward the end of the program. Her SNAP score also shifted from 0.33 to minus 2.58, which may again indicate great recognition of cognitive or emotional challenges after engaging in structured tasks and reflection. The SNAP questionnaire, I have to mention that is a questionnaire that we have designed in our lab in order to capture pre and post situation in neurocognitive symptoms in COVID conditions. We have a poster about SNAP, and you can come to discuss about it. And now I will pass the stage to my colleague Maria Loizio to talk to you about the preliminary neuropsychological outcomes in the assessment of post-COVID-19 brain findings.
Thank you, Flora. So my name is Maria, and together with my colleague Galia, we will share a bit more about the findings of the project in terms of the neurophysiological measures. So as my colleagues have mentioned, we used FNIRS as part of our baseline assessment. So that is functional near-infrared spectroscopy, which is a non-invasive brain imaging technique that measures the concentration of oxy and deoxygenated hemoglobin by using infrared light. So we used a flexible sensor pad, just like the one you see on the slide, with 16 optodes that function at a 2 Hz frequency. And we applied this pad on the dorsolateral prefrontal cortex, so at our participants' foreheads, because this area has been shown to be involved in executive functioning tasks. Our participants were asked to respond to a computerized executive functioning task that was developed in our lab. Basically, they were given 4 possible scenarios with 4 possible responses each, and they were asked to indicate how likely they were to act in a certain way by rating the scenario on a 4-point scale. So our aim with this task was to measure the levels of oxygenation using the FNIRS machine during this executive functioning decision-making process. So after filtering and pre-processing of the raw FNIRS data, we collected information on oxyhemoglobin, so the amount of oxygenated hemoglobin in the area, the deoxyhemoglobin, the amount of oxygen-depleted hemoglobin, the total hemoglobin, which measures the overall blood volume changes in the region, and the differential hemoglobin that measured the change in the total hemoglobin. Our participants also received an MRI scan as part of the baseline assessment and prior to the cognitive intervention. So our MRI images were pre-processed on SPM, which involved normalizing the image to MNI space, segmenting the tissue in gray matter, white matter, and cerebrospinal fluid, doing some quality control analysis and smoothing to make sure that the images were appropriate and ready for the processing. In terms of the processing, we developed Python scripts to extract regions of interest for all the participants and enter those on a centralized database. And then we ran another script to assess the Pearson's correlations between those regions of interest and the FNIRS data that we extracted. And now I will hand over to my colleague Galia, who will talk a bit more about the data analysis process. Just a second, please. Sorry about that. Okay, so the objective was to examine how brain structure measured with MRI relates to functional activation, measured with the FNIRS, both at rest and during the cognitive task. We ran cross-modal correlations between regional brain volumes and hemodynamic activity, applied the false discovery rate, the FDR correction to control for multiple comparisons, and then extracted and visualized only the significant brain behavior associations. We found a strong positive association between the left ventral diencephalon gray matter volume and the HBO, the oxygenated hemoglobin levels during the executive task at baseline. Additional associations appeared in frontal and subcortical regions, showing that both areas work together through coordinated neurovascular activity even before the intervention. On the graph here, you can see the median and the interquartile range for both deoxygenated hemoglobin and the total hemoglobin, comparing the pre- and post-intervention measurements. Statistical analysis using the Wilcoxon test showed no significant pre- to post-intervention differences. However, when we looked at the effect sizes, both deoxygenated hemoglobin and the total hemoglobin showed large effects, which suggests that while these changes weren't statistically significant, they may still reflect meaningful physiological increases following the active stimulation. Moving on to the between-group comparison of the task-related changes in the total hemoglobin between the active and the non-active TDCS groups, you can see the mean and the standard error for each group with individual participant data points shown in black.
The Mann-Whitney test indeed indicated that the difference between groups was not statistically significant. However, similar to the previous slide, we observed a large effect size, which suggests a trend toward greater task-related related increases in the total hemoglobin for participants who received active stimulation compared to those in the non-active group. So in this analysis, we see that individuals with PCS appear to rely on neurovascular responses that are less efficient, which likely requires greater recruitment of frontal subcortical networks to maintain performance. This gives us a clearer picture of the mechanisms that may underlie the cognitive fatigue we observe clinically. These findings also point directly to the next steps, comparing these structural hemodynamic relationships with healthy controls and evaluating whether targeted interventions can modulate or normalize these patterns. And now back to Professor Constantinidou for the concluding remarks.
Okay, lots of information, right? So let's just try and put it all together. All right, so what do we know so far based on our studies? In terms of the cross-sectional data, about 90% of the 416 participants had at least one infection. The median number of infections was 2. The median time since last infection at the time of the study was 19 months. 30% of the sample reported lingering symptoms resulting from COVID-19. Brain fog, headaches, physical fatigue, and neurological fatigue, and body aches are the most prominent symptoms in this cohort, and this aligns with international literature. In terms of cognitive performance, age and education, as one would expect, were the most robust predictors of cognitive performance based on the tasks we implemented, the cognitron. The number of COVID-19 infections was predictive of cognitive performance. So this aligns with Dr. Nikolopoulos' pitch that vaccination and prevention is important. So the more infections we sustain, the higher the risk of having long COVID and reduction in cognitive performance. Psychosocial health, as measured by the study measures, was not predictive of cognitive performance for this particular sample. Now, how about the feasibility and the effects of the intervention? For those enrolled, adherence was strong. So once we were able to convince people to participate, they stayed with the project. Most participants completed the intervention, reporting it as relevant and beneficial, using multiple measures, using the feasibility satisfaction questionnaire that Dr. Solomou presented, qualitative questionnaires, and then objective data from the cognitive intervention. Participants described a sense of cognitive clarity and improved word finding in daily life. The therapeutic rapport emerged as a key motivator for engagement and completion, emphasizing the trust and collaboration with the therapist, and that was central to maintaining motivation and effort throughout the sessions. Pre-post gains on CP tests across the whole sample, regardless of the NIPS condition, suggest that the cognitive training alone is producing meaningful improvements. Both sham and active TDCS groups appear to maintain cognitive function, with pre-post analysis showing a trend towards improved MMSE. But I would like to alert us to that, because even half a standard point or half Z-score reduction in performance for those who are functioning highly may be an effect of a significant effect in their daily performance. But in terms of numbers, we didn't see statistical neurodegeneration. Many participants presented hypervigilance regarding health, symptoms, frequently monitoring both physical and cognitive sensations. This is a known feature of the post-COVID related anxiety. In some cases, TDCS produce mild discomfort, likely amplifying this vigilance, which may have influenced anxiety or depressive responses and discontinuation in some instances. Interestingly, qualitative reports showed a strong subjective perception of improvement and benefit, even though was not reflected in the PHQ and the anxiety measures. So there might be a disconnection between the standard screening tools that we use in projects and subjective experience, or that the psychosocial tests that we used lack the sensitivity to capture subtle changes in symptom recovery. Now, in terms of our biomarkers, FNIRS is a biomarker, and the MRI biomarkers of PCS implemented in this study. Associations between subcortical gray matter volume and hemodynamic activation was present. Participants with long COVID appeared to show baseline compensatory neurovascular activation during cognitive tasks, especially those requiring prefrontal activation, like executive functioning tasks. However, further research is needed, of course, to clarify these mechanisms and validate FNIRS as a biomarker in this population. Although no statistically significant group differences were detected between the sham and the active TDCS, large effect sizes in HBR and total hemoglobin indicate meaningful physiological increases following active stimulation. And these patterns suggest promising neural engagement and support the potential FNIRS measures as a sensitive biomarkers of brain response due to non-invasive brain stimulation. Now, what are some implications about future research directions? Of course, we want to increase sample size, and one of the challenges being in a small country like Cyprus is that our clinical studies often have small sample sizes. But future research needs to continue to explore the cognitive intervention as it shows beneficial effects, supporting its role in addressing post-COVID neuropsychological symptoms. Evidence in our project suggests two distinct neuropsychological phenotypes in PCS, a predominantly affective phenotype and a predominantly cognitive phenotype, and we need to explore this hypothesis and findings and trends better. There was a maintenance of cognitive performance and no evidence of a neurodegenerative phenotype among participants under 65. For participants who are older, maybe that's a different pattern. Future research should integrate psychosocial intervention alongside with the cognitive and NIPS intervention to target the multidimensional symptoms of PCS. There is a need for more sensitive psychological measures to fully capture the affective component of PCS, and efficacy data must be established before advancing toward precision rehabilitation in post-COVID symptoms, and larger RCTs are needed. For us in Cyprus, we have significant referral pipeline limitations in that in Cyprus we have major challenge for sustaining clinical research in this population in order to have a representative sample because the long COVID clinic at the Nicosia General Hospital, which was the main clinic, it's really not receiving referrals anymore from physicians, and therefore it seems that patients are not really identified with PCS. We know they exist because we know Cyprus continues to have infections, so we know they exist, but especially for mental health and cognitive health, we need to do better work in terms of identifying and helping patients seek support, and the panel this afternoon will also address this issue. So with that, I would like to really thank the entire team. These are key people on the team from iLab at the University of Cyprus, collaborations from the other University of Cyprus institutions, Maastricht University, Ghent University, and King's College. So thank you. I'm very mindful of the time that we're cutting into coffee, and I know we need coffee to continue activation of neurons. So we'll take one question, and then we'll take more questions when we're out. So if anyone has a very burning question, then.
First of all, thank you so much for this talk. I think this is a very relevant and urgent topic. My question is more about the effects of COVID, not just of COVID inherently as a virus, but as the effects of the measures that we took. So, for example, there are some reports that reveal that domestic violence rates increased with quarantines. How can we prevent an effect like this happening for future pandemics?
Mm-hmm. So I'm not an expert on policy, but certainly I know at the same time that we were conducting this study, Flora was conducting her doctoral dissertation, which was actually, you want to talk about your, yeah.
Actually, I was conducted my PhD dissertation just after or during COVID, with collaboration with the NGO in Cyprus that handling cases of domestic violence, and indeed, we found that the cases of domestic violence increased during COVID, and the main thing that we should do in order to prevent that happening, it's more policies against domestic violence, because we can't move people outside from their home in order to not get domestic violence, but we have to create new policies and more awareness about domestic violence in order to prevent that from happening in the future. The home of someone should be the safe place to live, so that's very important, yeah.
But thank you, Flora, but you're also raising the important issue, what were the effects of COVID and isolation measure? And I think it goes back to what Dr. Nikolopoulos talked about. You're trying to, when you're implementing policy in a crisis, because it was a crisis, right? You're trying to look at multiple variables and make decisions as a system in order to manage the epidemic. And hopefully, not just for violence, but including violence and other things that we saw that were negative effects of COVID. So hopefully health systems have learned, and that if anything similar happens, there will be some systems in place to help the public navigate through these difficult times. So thank you. Okay.
Thank you very much to Professor Constantinidou and the Neurocognitive Laboratory team for this comprehensive and insightful overview of the BRAIN project. Our sincere thanks also to Professor Yorgos Nikolopoulos for his valuable contribution. Thank you, Professor Zelaya, for your informative and thought-provoking presentation. Ladies and gentlemen, distinguished guests, we will now have a 20-minute comfort break right outside these doors. In the main hallway, you will find the coffee, pastries, and our poster exhibition. The director of the Center for Applied Neuroscience, Professor Fofi Constantinidou, will coordinate the remainder of the scientific program with our distinguished speakers. I wish you all a productive and inspiring continuation of the conference.
I'm back. I hope you enjoyed the coffee break. I hope you had the chance to see the posters of the researchers and the research teams working here in collaboration with the Center for Applied Neurosciences mostly, but also other local teams. We're back here for the short presentations for which the selection committee selected 5 presentations covering several topics in the field. All presentations will happen consecutively. So you're kindly asked to hold your questions and queries for the last part where everyone is finished, where we will address all of them collectively. Now we can begin with the short presentations, and I would like to ask the presenters to come on stage, all of them. That will make easier the whole process. Okay, so thank you. And without further ado, the first Blitz presentation will be delivered by Efrosini Charalambous, who is a postdoctoral researcher from the Onissilos Co-Fund project, MSCA fellowship actually. And the floor is yours.
Okay. Good morning, everybody. So the topic of this presentation is the age and environment attunement and the dimension of affectivity. It is a part of the co-funded project by the European Union Horizon 2020 and the University of Cyprus. And the project, the project explores how the dynamics of the social and material environment shape emotion and situated affectivity. By treating rhythm as a way of connecting brain, body, and environment, it brings together relevant research and theories to identify future directions for research and architectural design as well. So to start, a very brief introduction, theories from ecological psychology and from enactive cognition provide frameworks for better understanding how we attune to the rhythms of the built environment. Unlike traditional approaches, these perspectives, they emphasize the role of the environment in perception, cognition, and behavior. So brain and body rhythms, they often couple according to specific principles, and they form a hierarchical system that influences how we perceive and evaluate our surroundings. Slower body rhythms, like the heartbeat, according to evidence, they influence faster brain oscillations. And together they create a temporal mechanism that tracks changes across different timescales. According to a recent perspective published this month, this rhythm organize our experience of time, and they are also sensitive to emotions. Arousal, for example, can accelerate or slow the heartbeat, expanding or contracting subjective time. This process enables the synchronization with external rhythms, so it's about adaptability. Additionally, evidence suggests that the neural oscillations and behavior align with environmental rhythms. Findings show that people tend to slow down when the rate of change in the optic flow increases. This demonstrates how external patterns can shape brain and body coupling. This suggests that the rhythmic nature of body and brain interaction can dynamically couple with environmental rhythms, which fosters experiences of natural attunement. A central idea of direct perception,
that has its origin in ecological psychology is that the perceptual system simply extracts the invariants from the flowing array, such as the optic flow. It resonates to the invariant structure, or it attunes to it, as Gibson comments. Attunement and resonance and entrainment are all related to properties of dynamical systems. Resonance is a fundamental property of perceptual and neural systems operating across multiple scales. Entrainment has a unidirectional character and is defined as a temporal alignment of oscillatory activity to a driving external rhythm. They often support this experiential quality of attunement, which is the sense of connection of self-tuning to environmental variables, which carries a more intentional connotation. So relational structures, such as affordances, behavioral settings, and atmospheres, can help us better understand the relationship between situated affectivity and the design of the built environment. Affordances are the opportunities for action offered in the environment, while behavioral settings are higher order structures with spatial and temporal boundaries. They support structural patterns of coordinated activity, behavior, that invite and sustain an individual's engagement. Affective atmospheres, they provide a shared ground from which subjective states and emotion emerge. They shape and invite an individual's affective engagement, and they support attunement and anticipation of events. Affectivity, thus, can be seen as a relational process of sense-making that takes place between the agent and the environment, or the agents and the environment. Concepts like affective scaffolding, built on the idea of situated affectivity and situated cognition, and show that emotions are dynamically co-regulated by external environmental resources. So the ability to regulate affective states by changing one's environment is linked to the concept of niche construction, which refers to how an animal lives rather than to where it lives. In this view, affective niche construction involves material elements, practices, social groups, and cultural structures that shape and organize our affective life. So several themes emerge from bringing together these insights and point towards potential future directions for exploration. The concept of attunement, atmospheres, and affective scaffolding foreground affectivity as a dynamic process rather than a static property of environments or individuals. This extends current critiques and challenges traditional static views of architecture. It draws attention to its temporal unfolding, opening pathways towards a more process-oriented understanding of the affective experience of architectural space. Moreover, active engagement with the affective atmosphere of a theatrical performance, for example, or a music concert can modulate bodily rhythms. Studies of audiences at classical music concerts have shown synchronization in signals like heartbeat, breathing, and skin conductance. These findings reveal how bodily rhythms, which in turn influence brain rhythms, intertwine with environmental structures to create a dynamic fields of attunement. These collective synchronizations illustrate how atmospheric rhythms extend beyond the individual, shaping shared affective experiences that unfold over time. So concepts such as rhythm, vibration, and tonality help explain how constellations of element and energy patterns shape affective atmospheres. The concept of vibration shifts the focus to elements that function as thresholds, influencing a body's or an object's capacity to act. A nice example is the urban carpet, which is a simple installation, a woven textile with charred wood at its crossroad. It draws attention to the sensory experience of touch and resistance that you feel when stepping on it, and smell. This shift in experience promotes spontaneous actions like foot tapping and dancing. This shift influences the action is linked to the phenomenon of metastable attunement, that is the flexible balance between stability, openness to novelty, in age and environment coupling, and enables a switch into new behavioral patterns. However, differences associated with how we switch to new behavioral patterns are associated with personal tendencies, habits, or broader social and cultural and gender dynamics that shape how we attune to affective atmospheres. They may cause specific individuals to feel disoriented or excluded in particular environments. such as, for example, neurodivergent individuals may experience difficulties tuning in and orienting in environments that are noisy or brightly. So designing for neurodiversity or diversity means recognizing that some settings may not accommodate different styles of bodily being in the world, limiting social connection and the feeling of being at home. Thank you.
Thank you, Efrosini, for your talk. And now I would like to welcome Evi Kostadinidou, a researcher at the Department of Psychology, for her presentation. Welcome.
Okay. Hello, everyone. My name is Evi Kostadinidou. The title of my presentation today is an experimental study on the impact of survey stakes on response inconsistency in mixed worded scales. This is a collaborative work with Michalis Michailidis from the Department of Psychology here at the University of Cyprus. Surveys are widely used in psychology and education, and they can measure traits, attitudes, behaviors, demographic experiences, emotions. One of the challenges, though, that we encounter when we deal with survey data has to do with the careless or insufficient effort responding, because this behavior can reduce data quality and biases research conclusions. So the focus of this study is on inconsistent responding, especially we focus on answers that do not logically align across related items in mixed worded scales that are included in surveys. The main aim of this study is to investigate how increasing perceived stakes affect inconsistent responding in low and high stakes conditions. So we tested three hypotheses. With the first hypothesis, we expected more inconsistent responding in low stakes. With the second hypothesis, we expected the response times to be longer in high stakes. And with the last hypothesis, we expected that conscientiousness will predict lower inconsistent responding in low stakes conditions. In this study, we followed a within participant design with 66 students, mostly from the Department of Psychology here at the university. The study used high and low stakes conditions in a counterbalance design. And for our analysis, and for the identification of inconsistent respondents, we first used the mean absolute difference approach. Then for the comparison of the response times between the two conditions, we ran the paired sample test, and we also tested a negative binomial model for the prediction of inconsistent responding. Regarding experimental manipulation, we provided specific instructions for both conditions, the high and the low stakes conditions. Regarding our results and for the prevalence of inconsistent responding, we used two criteria. And through the three criteria, we identified fewer inconsistencies in high stakes, but the differences between the two conditions was not statistically significant. However, when we used the more lenient criteria, we did find more inconsistencies in low stakes, and the difference between the two conditions was statistically significant. And at the scale level, we found those differences in the Rosenberg scale and in the self-regulation scales. About our second hypothesis and the response time, we found significantly longer response times in high stakes in all of the 5 scales included in both conditions. And this supports the idea that increased the stakes linked to more deliberate responding. About our last hypothesis and the role of conscientiousness, we found that conscientiousness was an important predictor in low stakes, but not in high stakes, as you can see here at the table as well. And this can be supported also from previous studies that say that conscientious people tend to be more careful, so we do not expect to be inconsistent in their answering. And in conclusion, we found slightly more inconsistent responding in low stakes through differences between the two conditions were small and not always significant. About the response times, we found that participants took significant more time to respond under high stakes conditions, supporting that increase the stakes enhance participants' engagement and deliberation. Also, we found that higher conscientiousness predicted lower inconsistencies in low-stakes conditions. And about the manipulation check, we found that participants were more focused. This is what they said through the self-report in high stakes, but compliance was high in both conditions, considering the instructed item that was included in both conditions, in both questionnaires. Thank you very much.
Thank you very much, Hevi, for the presentation. And now I would like to welcome Dr. Anna Onisiforu, who is a senior scientist in the systems AI and systems bioinformatics unit of the translational neuropsychopharmacology, supervised by Panos Zanos.
Hi everyone, my name is Anna Onisiforu. I'm a senior scientist and head of the
Hello, everyone. My name is Dr. Anna Onisiforu. I'm a senior scientist and head of the AI System Bioinformatics Unit at the Translational Neuropharmacology Laboratory, known as Zanos Lab, at the University of Cyprus. My research focuses primarily on investigating microbial pathogenesis in neurodegenerative autoimmune diseases. This is a work I started during my PhD work, where I conceptualized and developed novel integrated bioinformatic methods that allow to infer viral influence in the development of neurodegenerative diseases. And then subsequently, I expanded this work currently now. And today, I will present you one of our latest papers, VTRAC, a framework for inferring viral influence on disease-associated transcriptomes that we apply in the case of EPV and multiple sclerosis. So Epstein-Barr virus affects 90% of the global population, which means that here now 9 out of 10 people have the virus. Circumvention of EPV increases 30% of the time of having multiple sclerosis, and MS patients have elevated EPV-specific antibodies and autoimmune responses to latent EPV antigens. There are several hypotheses about how viruses can lead to autoimmune diseases like MS. The most prominent one is through molecular mimicry or dysregulated EPV antigens and through viral-host interactions. So MS is an autoimmune demyelinating disease of the central nervous system. It has 4 clinical types: clinical isolated syndrome, relapse repeating MS, secondary progressive MS, and primary progressive MS. So the relapse-remitting phase of MS is characterized by inflammation, and its pattern resembles a lot of the reactivation of viruses. Infection with a viral infection can increase the risk of a relapse, whereas the primary progressive and secondary progressive have pure response to immunotherapies and characterized by neurodegeneration. So the question that we try to answer is how EPV affects across MS clinical types. To answer this question, we created an integrative approach. We first analyzed MS blood samples of transcriptomic profiles of MS patients across the 4 clinical types and found differentially upregulated and downregulated genes for each type. And then we also collected experimental viral-host protein interactions and integrate them into create EPV host MS clinical type-specific protein interaction networks, one for each type. We subsequently extracted from each network the upregulated and downregulated differentially expressed genes that are targeted by Epstein-Barr virus in each type. We also analyzed the network using topological analysis to identify and rank viral proteins according to the ability to target proteins in the networks, which are high centrality nodes, human proteins that interact with other proteins that enable the virus to exert systemic pathogenic effects. We also perform enrichment analysis to identify the functional groups and biological processes that are modulated by Epstein-Barr virus across the 4 clinical types. So here we can see the results that indicate the upregulated and downregulated differentially expressed genes targeted by EPV across the 4 MS types. And we can see that it mostly targets upregulated differentially expressed genes, particularly in the disease relapse remitting and primary progressive, whereas in the secondary progressive, it primarily targets downregulated genes. And here we can see the exact viral proteins in each stage and how many differentially expressed genes they target. And we can see that BFVLF1 viral protein, which is involved in the viral reactivation of the virus, targets a lot of differentially expressed genes in the early stages. And here we can see the ranking of the viral proteins according to how they target proteins in the networks across the 4 clinical types. And we can see EPNA-LP protein, which is involved in the immortalization of B cells, the reservoir where EPV establishes latency, ranks first in targeting high centrality nodes in the network. We also perform enrichment analysis of the differentially upregulated genes, and we found that in the early MS, EPV targeted upregulated B cell-related processes, toll-like receptor signaling, and infection-like inflammatory pathways, while suppressing antiviral responses. Whereas in the progressive clinical types, it exhibited fewer viral connections but distinct mechanistic shifts. For instance, in the secondary progressive emitting phase, it was suppression of vascular and cardiac repair-associated pathways, whereas in the primary progressive was dominated by upregulation of vascular and lysosomal remodeling processes. So overall, EPV exerts clinical type-specific perturbations with strong immodulation in early disease and selective cellular remodeling during progressive type. So VTRAC framework allows to identify key viral proteins and host pathways for clinical type-specific tailored therapeutic targeting in MS. It also offers a general basal systems-level framework for elucidating viral contribution across complex diseases, including neurodegenerative diseases, autoimmune diseases, and cancer. So our research has been funded since 2023 by the Infectious Disease Society of America. And we also recently published another paper, VTarget, which allows to evaluate whether MS immunotherapies reinforce or reverse Epstein-Barr virus-driven dysregulation in MS. If you're interested to look it out, it's available in Virus. Thank you very much.
Thank you, Anna, for a great presentation. Now, the next presentation is organized by Panagiotis Papetas and Eleni Georgoudi, PhD students at the Neurocognitive Research Laboratory, which is supervised by Professor Fofi Konstantinidou.
Hello everyone. Today I will present our study titled Normative Data and Psychometric Evaluation of this Executive Questionnaire Revised in a non-clinical adult sample, associations with mood and post-COVID cognitive symptoms. Higher order cognitive processes such as self-control, flexibility, inhibition, planning, etc. are referred to as executive functions. These functions are extremely vulnerable to neurological, psychiatric conditions and infections such as COVID-19. Because of this, accurate measurement of everyday executive functions becomes crucial. And this executive questionnaire is a 37-item questionnaire that assesses everyday executive functions. However, most studies using DEX were based on English-speaking samples, while research in Greek populations remains limited. Therefore, our study sought to validate this questionnaire in Greek-speaking non-clinical adults under the age of 60, using and examining variables such as depression, anxiety, brain fog, sex, age, and the impact related to COVID-19, and whether these variables predict executive dysfunction in this population. We had three hypotheses. Younger adults would have higher levels of executive dysfunction. Higher DEX scores would be predicted by mood, brain fog, COVID impact, and female sex, and DEX will demonstrate good test-retest reliability over a 10-day period. 453 of the 467 adults from the general population who initially comprised our sample met the eligibility criteria, resulting in a final sample of 342 individuals after processing missing data. Majority of participants were female with a mean age of 31 years. We divided the sample into three groups to examine the age-related effects, and in addition to the brain fog scale, the PHQ-9 for depression, GND7 for anxiety, and the SNAP COVID tool, which measures symptom severity and the degree to which these symptoms were specifically associated with COVID-19. We also administered the Greek version of DEX, which measures everyday executive difficulties. Reliability was assessed by asking 79 participants to complete the survey 10 days later. We conducted ANOVA with Bonferroni correction and hierarchical regression. While the second model of regression included brain fog, COVID impact, sex, and age to assess their additional contribution to executive dysfunction, the first model examined whether anxiety and depression predicted DEX scores. Age differences were evident. In every comparison, younger adults reported higher executive difficulties. Test reliability over a 10-day period was generally good. Almost half of the variance in executive dysfunction was explained by depression and anxiety in the regression models, and this variance increased to 62.6% when brain fog, sex, age, and COVID impact were included. Brain fog was the strongest predictor, followed by mood. COVID impact and age had no distinct effect. Overall, emotional distress and subjective cognitive complaints, despite demographic characteristics, appear to be the main causes of everyday executive impairments in healthy individuals. According to our findings, younger adults reported higher executive difficulties, possibly as a result of stress and developmental variables. Depression, anxiety, and especially brain fog were the strongest predictors of everyday executive dysfunction. This is consistent with previous research showing that mood disorders can affect cognitive flexibility, and experiencing brain fog can negatively influence executive functions. Sex differences has moderate significance, and age-related effects, as well as COVID impact, were not found to be significant predictors. Furthermore, DEX showed strong test-retest reliability. Due to the cross-sectional design, the majority of sample by women, and the use of self-report measures, these results should be interpreted with caution. Overall, the study emphasizes the importance of mood symptoms and subjective cognitive complaints, and provides useful normative data for the Greek version of DEX. Future work should use longitudinal designs and combine performance-based executive function tasks with self-report measures to clarify changes after COVID-19 and assess whether interventions targeting mood and metacognitive awareness improve executive functioning. Thank you.
Thank you, Panagiotis. And now I would like to ask Margarita Zachariou on stage. Margarita is an associate scientist in the Cyprus Institute of the Neurology and Genetics and serves as a PI of the Meta Brain. Welcome.
Thank you, and thank you for including us in today's very interesting program. So I'm here with a slightly different talk today in terms of presenting a program that we are currently working on in the Cyprus Institute of Neurology and Genetics. We are based in the bioinformatics department, and Meta Brain is an European program that we're working on. And it's an 8-partner, 5-country size, It runs for 3 years, and it's very, very multidisciplinary. Includes engineers, clinicians, experimentalists, computational scientists, and so on. The main objectives are to develop and validate magnetoelectric nanoparticles towards modulating the brain activity. Also to study the stimulation effects, of focused ultrasound, and at the same time create the infrastructure, so then devices that can measure electrical activity in settings like a magnetic field, for example. And what we are also working on, develop the computational models that provide the mechanistic understanding of how these non-invasive stimulations affect the brain. So we're leading the work package on multi-scale models. And multi-scale here refers to the fact that we're trying to understand how changes in the cell reflect to changes in the network activity. And in the middle, we have the neurons that we model with equations. I only have one equation here, but you can think of many, many equations behind that. And we model the response of the cells, the effects on the cells, and the effects on the networks. And here we can see the effect on a slow oscillation. For example, how a magnetic nanoparticle intervention scheme resulted in increasing the frequency. This is not only for illustration, this is actually part of our preliminary results. Now, what we expect as impact from this program, as I said, it's a multifaceted sort of approach. So on one side, we expect that we will have a better idea on how to use these methods effectively, and the how here I mean to use them safely and precisely. I don't need to preach to this audience the value of non-invasive stimulation, right? But as we know, non-invasive can be not very focused. So this is where this program is trying to contribute really with these approaches. Of course, the end goal is to be able to control the activity, either increase or decrease, depending on the clinical scenario that we are implementing it. So in epilepsy, for example, we will need to decrease frequencies, firing rates, whereas in other diseases like Parkinson, we might need to increase. And since we're also in the process of modeling these setups, let's say, we're trying to get to the mechanistic explanation of how these stimulation protocols work, so we can better tailor them for different patients, for different diseases, and so on. So towards personalization as well. So that's all from us. The team that is working on this project and on other projects spanning from genes all the way to policy is with us here today. We have Stella, Caterina, Agis, and Rafael. And it's also a very multidisciplinary team, but this is what we really need for challenges these days. So very happy to be working with them. Thank you.
So thank you, Margarita. And this concludes our Blitz presentations. We have a few minutes for questions that can address any of the presentations, all of them collectively. Yes, please.
Can I ask about the... Order or punishment? What is actually... Yeah, because you talked about filling out these questionnaires and then you make it a high-stakes situation and a low stakes, and I didn't know whether that was... What is it high stakes or is it punishment?
No, we stick on the specific instructions that we gave to the participants. So for the high stakes conditions, we told them to behave and answer to the following questions. I don't remember exactly how we say that, but the main point was that they had to keep in mind that these questions will affect, the answers will affect, for example, their grade or their job. So it's important to, you have to be engaged in order to answer to these questions. But in the other condition, we write something that it was totally the opposite, that it will not affect your personal or your professional career, for example. So
I was
you
just
will
intrigued.
answer as
yeah.
you do in any survey.
I was just intrigued whether someone responds better to a high-stake reward than to a high-stake punishment. So whether there's the compliance and reaction times and everything would be equal.
The reward was for all of them in order to make them come and participate in our research. So they could choose between a credit or in a particular class or a voucher. So all of them would get something.
Thank you.
But these two issues took us a lot of time to decide and understand, because in the literature there are many approaches and you need to consider a lot of things in order to decide what exactly you will do.
Thank you very much.
Thank you for your question.
Any other question? Out there? Yeah, Sophie.
This is for Margarita. Can you tell us a little bit about the stimulation, like the depth? I mean, how deep are you able to guide the stimulation?
Right, so because we have started with slices, so the full brain experiments are taking place now, and this is in small animals, right? So like rats, for example, that I'm guessing it's for the focused ultrasound. So because the probe is rather large compared to the animal brain, now it takes like maybe 70% of the brain. So we're not at the stage of applying this yet to humans yet.
Thank you.
Thank you.
Any other question? If not, I also have one for the ultrasound. Now, when you apply radiation stimulation for cancer, for example, you basically target a region from multiple different sources or multiple different locations, and the activity sums up at your target. This actually can be performed with ultrasound as well. So you can send waves from different angles, and they could all sum up at the specific location. Is this the approach, or do you use one source of waveforms and just targeting and going through the complete tissues up until the target?
So I think for the ultrasound, I better redirect the question to our specialist for the ultrasound.
Okay. Well, in other disciplines, it is a standard practice. It could work with waveforms as well. Just an idea. Okay, so thank you very much. Thank you for your presentations. And we can move on to the next session. Welcome Professor Spanoudis to take the stand.
Okay, hi to everyone. Thank you for your attention during the Data Blitz presentations. We will now continue with the CAN presentations, showcasing the work of our research teams. So, given that we are running late, let's move very quickly and welcome Evgenia Peristera-Kouki to the podium. Evgenia will present on behalf of the Language and Cognitive Development Research Group and the Learning Disabilities Group of CAN She will present the title. Let me say also that given that we are running late, it's better to keep your questions for the end of this session. So please, Eugenia.
Just a minute. Perfect. So, hi from me too. I'm Evgenia Kouki, and today I will present to you our recently published work from the Learning Disabilities Lab. It's co-supervised by Professors Timothy Papadopoulos and George Panoudis. And it's entitled Single Deficit, Comorbidity, or Varying Degrees of Dysfunctions, New Directions to the Study of Learning Disorders. So to start, neurodevelopmental disorders such as specific learning disorders, specific language disorder, and attention deficit and hyperactivity disorders have severe and diffuse impact on both the individual as well as the family, school, and society as well. So, to conceptualize neurodevelopmental disorders, early literature has proposed that there are specific one-on-one correspondences of vulnerabilities from the etiological or genetic level up to the behavioral level. So they assumed that there is one specific core deficit that is reflected from the etiological level to neural circuits, cognitive deficits, and finally, they are reflected in the observed behavior. However, this is a very difficult hypothesis to actually confirm with what we know, as various deficits can coexist in one disorder, in more than one disorder, as well as one disorder can be characterized by many possible deficits. So from what you can see here, there have been also put forth several hypotheses over the last decades that have tried to stay faithful in this single deficit model, but have failed. Why is that? Because we have moved on to the multiple deficit model a few years later, which can account for interactions and not just causal relations between and across the various levels of analysis. And based on this rationale, the research domain criteria has extended the multifactorial work that has been done over time, and it has been proposed in the early 2010s. It is agnostic to categorical diagnosis, very much like the multiple deficit model. However, it also adds more levels of analysis and therefore more correspondences and thus possible pathways for psychopathology to be accounted for. So this can further extend neurodevelopmental work to actually explore etiologically founded classification schemes. And this is what we also have done in this systematic review. So, we explored the following questions. First of all, can the research domain criteria be used to identify specific subtypes of these neurodevelopmental conditions? If so, can it account for the overlaps and distinct pathways of these subtypes? And can it correspond these specific overlaps with neurophysiological findings? Finally, we also wanted to explore whether the RDoC framework can be validated through the results stemming from answering the previous questions. So, this systematic review explored published research from several databases that have been published in the last 10 years. We started from 2015 because we wanted to have, let's say, the first proposal that was RDoC related was in 2013. So we left a few years so that the actual related research can be published and peer reviewed. So that's all for our methodology. We have screened 2,000, I think it's been 3,000 studies in the first steps up to 2025, and we have come up with 15 studies at the end. So from what we see here, it's been a very limited time to actually delve into RDoC through primary research, but the results are interesting. First of all, as we see here, attention shows the research that targeted attention showed heterogeneity in the neurodevelopmental disorders in terms of how the deficits have been exhibited in several neurodevelopmental conditions. This has been replicated in the construct of perception, language, cognitive control, working memory, as well as processing speed. So I want to be brief, so I will just summarize all the forest plots and say that the heterogeneity indices, they are all significant, which shows that there is heterogeneity within the neurodevelopmental disorders populations. And the actual effects are non-significant, which means that when we use categorical diagnosis, we cannot really account for effect sizes within that will show causal relationships between categorical diagnosis and cognitive deficits. Let's move on to more results. Regarding the first question, it was shown that classifications based on general cognitive performance or on constructs, as shown in the forest plots, of cognitive control and working memory only partially explained the presence of neurodevelopmental conditions. Also, regarding the second question, no single deficit fully accounted for the range of deficits observed across conditions. There were domain general factors such as attention, working memory, and processing speed that were significant predictors across the various neurodevelopmental conditions. Broader difficulties overlapping conditions also were associated with more severe deficits. And these findings suggest that neurodevelopmental conditions are best explained when approached on a continuum. Regarding the third research question, inattention was associated with structural and neurophysiological abnormalities, and these abnormalities reflected early deficits in sensory processing. Language and memory outcomes were closely related to structural characteristics of the hippocampal regions, and also language development more frequently explored in terms of verbal IQ was linked to distinct patterns of activation in default mode network, in dorsal attention network, and salience network. Finally, the fourth question, the deficits underlying neurodevelopmental conditions were interconnected and interacted with each other, and the conclusions of this study align with other RDoC-informed findings and transdiagnostic research in general. So, to sum everything up, we want to highlight the importance of adapting a multifactorial framework such as the RDoC in understanding the neurodevelopmental conditions. And we think that the dynamic integrative approach can advance diagnostic procedures and inform interventions for individuals with neurodevelopmental conditions. Thank you very much.
Thank you, Eugenia. You were so punctual. Let's move now to the next talk and welcome to the podium Ioanna Kazalaki. Ioanna will present on behalf of Clinical Psychology and Psychophysiology Lab. The title of the presentation is Applying Emotion and Psychophysiology Methods to the Study of Cancer, Subjective and Physiological Markers of Emotion Regulation in Women with Breast Cancer.
Thank you very much. So, hello everyone. My name is Ioanna Kazalaki. I am a school psychologist and a PhD candidate in clinical psychology. So today it's my pleasure to present this research work that has been conducted under the supervision of Dr. Georgia Panagiotou, who is my supervisor in my PhD. So basically okay, let's open it. So basically, the topic that we are going to discuss has to do with emotion regulation, psychopathology symptoms, and HRV in breast cancer patients, how physiology with psychology are interconnected in breast cancer. So I will present you two studies that I completed with my supervisor. The first has to do with a systematic review that tried to summarize the findings that have to do with psychopathology symptoms such as depression, anxiety, and stress, and how these are linked with low HRV in breast cancer. I will explain why this is very important to research. And the second is a research study that was part of a larger project, which is called CARE, and was conducted in collaboration with other professors, Professor Zanos, Professor Maria Carecla, and others. So it's part of a large team. But before we start, I would like us to go through some theoretics so that we can understand better the studies that I will present. First of all, breast cancer is a type of cancer that originates in the cells of breast tissue and often originates from the ducts or the lobules, which are involved in milk production and transport. The symptoms vary based on the woman. There's a lot of heterogeneity, but the most frequent symptoms that we observe is breast pain, breast dimpling, or skin texture changes, lump in the breast, inverted nipple, change in breast shape or size, as well as redness. Approximately 1/4 of breast cancer patients present emotions such as stress, depression, and anxiety, which are part of a diagnostic category, generalized anxiety disorder, depressive disorder, and these rates are 5 times higher than the general population, so something is happening here. These emotions can influence, as we know from the literature, the autonomic nervous system, and more specifically, the cardiac vehicle control, which is linked to heart rate variability. HRV is the index that I am going to present you and talk to you about, as it is highly connected in low levels with cardiovascular disease, which is the main cause of death in breast cancer patients. Cancer is not the first reason why these women die, it's cardiovascular diseases. So we have to examine the mechanisms of why this happened in order to help these women live a valuable and good life. So HRV is a measurement of the variation in time between successive heartbeats and reflects the functioning of the autonomic nervous system. In high levels, high HRV reflects well-being and good psychological and physical health, as it shows that the body tries to adapt to the environment. In contrast, in low levels, we observe that the body does not react, does not try to adapt to the environment, and therefore, this is connected with poor emotional and physical health. Why do we care about this in the context of breast cancer? So basically, breast cancer patients present both, as I told you, negative psychological symptoms as well as low HRV. And literature has shown that these are elements that are highly connected and create a very poor quality of life in these women. So we see on the top the softer image of these variables, and on the down area we can see how excessive and difficult these variables can be transformed in later stages. So the systematic review has the title Innovating Findings on the Association between the Psychopathology Symptoms and HRV. And it was conducted in order for me to have a better knowledge on the literature so that I can understand what are the research gaps and fill them later on in the research study that we conducted as a team. So I used Prisma Statement. The databases that were included were Google Scholar, PubMed, and Scopus. The time frame was from 2000 to 2023. The language was in English. The sample was metastatic, non-metastatic patients and breast cancer survivors. We used many designs and we extracted and synthesized the characteristics of the participants and the studies that we included. And we used search terms that were related with the psychopathological symptoms, the HRV, and the emotional regulation. So this is the flow chart. As you can see, we concluded to 30 studies. So basically, 23 studies confirmed a negative association between HRV and psychological symptoms. So as the HRV would increase, these patients would present better psychological state, but on the other hand, when the HRV was reduced, the patients would present with depression, anxiety, and stress. 7 studies did not support this association, mainly in the early stage patients, and the most frequently reported symptoms from these patients were anxiety, depression, fatigue, distress, and fear of recurrence. For the last one, I can confirm that these patients have many thoughts that concern them because I had contact with them and they would frequently discuss about this. And the most commonly reported interventions had to do with the HRV biofeedback, mindfulness, yoga, music therapy, and others. So as you can see, I tried to create a table to summarize all these findings. The HRV parasympathetic indices would increase when these patients would be psychologically well. But, however, when it comes to the sympathetic indices or the indices that combine sympathetic and parasympathetic, here, as they would increase, the psychopathological symptoms would increase as well. So what to keep from here, that the more the parasympathetic system increases and works well, the better psychological state in this population. So HRV is a reliable biological marker of psychological status, and the relationship between HRV and psychological symptoms is longer in the long term. As we observed with my supervisor in survivors and metastatic stages. There is further research that has to be conducted in order to further understand these relationships. Now, when it comes to the empirical study that, as I told you, was part of the larger project, tried to fill these gaps in women who would receive the chemotherapy or radiotherapy. So these are data that are pre-treatment. So the primary aim was to examine whether subjective and objective indicators of emotional regulation through self-report emotional regulation ability and objective emotional regulation, which is reflected through HRV, are linked to symptoms of depression, anxiety, and stress in diagnosed breast cancer patients awaiting treatment. So we included 73 women, which was found to be enough through power analysis. who were diagnosed with Herceptin negative estrogen positive breast cancer and scheduled to receive these treatments. Why did we include this subtype of cancer? Because they would follow very similar treatment plan, which would help the research method to be as consistent as it could be. The materials that we used were questionnaires, demographics, and medical data were extracted from these. And we used the DARES questionnaire, which was about the subjective emotion regulation ability, DAS questionnaire, which was about depression, anxiety, and stress, and the HRV, which was measured for 5 minutes under rest. So we received the data from the questionnaires from the patients, and then we had the recording of HRV with them. I will not go through all of this. What I would like just to point out here, because I think it's very important and very consistent with the literature, is that in our sample, almost 50% of the breast cancer patients would consume alcohol daily. So this should create some thoughts. Now, when it comes to the psychopathological symptoms and emotion regulation in objective and subjective way, they reported mild psychological distress and slightly reduced HRV. And when we compared the treatment groups, radiotherapy and chemotherapy groups, we found no differences when it comes to HRV, emotional regulation ability, and psychopathological symptoms, as well as when it comes to other demographics and medical data. The only difference that we found was when it comes to cancer stage, which was increased in chemotherapy patients. So when it comes to correlations, we found strong positive correlations among depression, anxiety, and stress. This is something very... We understand why. Depression, anxiety, and stress scores were linked with greater emotional regulation difficulties, and low HRV was associated with higher distress in these women. All these align with the existing literature that we know. Also, we conducted hierarchical regressions to examine the predictors of psychological symptoms. In step 1, we had demographics. Then we added medical and lifestyle variables. Afterwards, emotion regulation subscales. And lastly, psychological physiological HRV indices. What we found here is that the type of treatment did not significantly predict levels of anxiety, depression, and stress. Difficulties in emotional clarity predicted higher levels of anxiety. So this tells us that the more clear the emotions that we have and the better we can label them, this predicts better psychological state. However, when it comes to emotional awareness, we found that the more aware these women were, the worse. And this may be similarly to the non-acceptance of emotion that was found in the subscale of non-acceptance. Maybe is a defensive mechanism here, but more studies will clear this. Low HRV, talking about the parasympathetic index of HRV was associated with higher anxiety, which also aligns with the literature, and low LFHF ratio, which is an indicator of the autonomic nervous system dysfunction, was linked to increased stress. To finalize, the last finding that we had was through moderated mediation that we conducted. Here, the HRV indices were the dependent variables. The independent variables were the psychopathological symptoms. As mediators, we included their subscales, and as moderators, type of treatment. In the case of LFHF ratio, we did not find any significant findings. However, in the RMSSD, we found that depression could predict RMSSD, and this was moderated by treatment type, which was significant in the chemotherapy treatment. And when it comes to stress, it predicted RMSSD and was moderated by treatment type again. And there's clarity predicted all psychopathological symptoms, but did not mediate HRV effects. So the key findings from that are that HRV shows reduced global autonomic regulation. So basically, we confirm what we know. Emotion regulation, especially clarity and awareness, is a central predictor of depression, anxiety, and stress. RMSSD, a parasympathetic index, is most sensitive to emotional differences and conceptual differences. Treatment type moderates relationships between distress and RMSSD, and we did not find group differences in emotion-related outcomes based on the upcoming treatment. So we had many limitations. I will just mention the fact that we conducted moderated mediation in cross-sectional data, which is not recommended. However, we expect for long-term and longitudinal studies to be conducted in the future to cover these limitations. So to conclude, psychological distress in breast cancer patients is closely tied to both subjective and physiological emotion regulation processes. Emotional clarity, emotional awareness, and parasympathetic flexibility, mainly when it comes to RMSSD, play central roles. And these findings can help us create interventions, both to target physiological and psychological elements to help these women have better quality of life, psychologiall state, less cardiovascular disease, and overall higher quality of life. So CBT, ACT, and all HRV biofeedback interventions are recommended. Hope for the best of this research, and thank you very much.
For your vibrant talk. Let's move now to the third team lab, and let's welcome Panagiotis to the podium, who will present on behalf of Cognitive Neuroscience and Functional Neuroimaging Lab. Their presentation is titled The Neural Substrates of Food Image Processing in Eating Disorders.
Hello, everyone. My name is Panagiotis Loizou. I'm a PhD candidate in the Department of Psychology at the University of Cyprus, and today I'm going to present to you a part of an ongoing research we are conducting with Dr. Evangelos Paraskevopoulos, which is my mentor and supervisor. I will try to be as brief as possible. So, regarding the theoretical framework of our study, it is based on three simple points that are very well supported in the bibliography, starting with the fact that anorexia nervosa patients demonstrated substantial cognitive and neural impairments when processing food stimuli. Additionally, they showed diminished interest in food and food images compared to healthy controls. Additionally, some studies supported the fact that anorexia nervosa patients perceive food stimuli as threatening. Now, regarding the aim of our study was to investigate the processing of food-related stimuli in anorexia nervosa patients, in healthy controls, and in overweight and obese participants, but also to identify and interpret potential cognitive and neural mechanisms that we will identify. Now, specifically, the questions that we wanted to answer for this part of the research was firstly to identify whether anorexia nervosa patients and healthy controls differed in the way they process food images. Also to identify if anorexia nervosa patients and healthy controls differed in the way they rated palatability, which means tastiness, of food images. And also to explore if anorexia nervosa patients process food-related stimuli the same way they process emotionally charged general images. Now, to answer to these questions, we used an integrated multimodal approach, which comprised of using multiple methods simultaneously, such as the EEG, such as the eye tracker, as well as yes and no questions as the behavioral part, where the participants had to answer if they believe or did not believe that the presented stimuli was tasty or else palatable. So the reason we adopted this approach was firstly because we wanted to address previous methodological challenges and limitations in the field, but also because we wanted to make sure that our datasets are rich enough in order to be able to meaningfully interpret and not only detect the neural and cognitive mechanisms during the processing of food images, of course. So regarding our design, we use a cross-sectional quasi-experimental design, and we had 3 groups. One experimental group that contained 17 anorexia nervosa or atypical anorexia nervosa patients, as well as 2 healthy control groups. The first group of the healthy controls contained 17 overweight or obese participants, and the second one contained 17 healthy participants. Of course, we made sure that none of the participants included in the control groups didn't suffer or had a history of eating disorder. Now, as you can see in the images, the stimuli we used were food stimuli and IAPS pictures. And we had 2 blocks. In the first block, we presented low-calorie and high-calorie food stimuli that were followed by a palatability question, which was basically asking the participants to answer using their a right or a left click of the mouse, whether they believed that the food presented was palatable or not. Sorry. The second block consisted of positive and negative IAPS pictures without any behavioral questions. Now, in order to address our first question, which was to identify whether anorexia nervosa patients and healthy controls differed in the way they process food images, we conducted sensor-level within-group analysis to see how each of our groups process high and low-calorie images. And as you can see in the charts, it's the strength of the EEG signal and how it changes over the time of the processing, of course, for each group. You can also see the statistically significant time windows, which are marked as gray, you can see, for each of the groups. Now, we chose to focus on a specific time window, you can see it in the pictures, that spans from the 388th millisecond until the 418th millisecond, which falls within the N400 because it appears around the 400th millisecond of processing, which we know that it reflects how we process meaning and how we make sense of things, basically. It's a top-down processing. And the reason we decided to focus on that time window, it is because it appeared to be statistically significant in each of our groups. Now, by using exactly the same time window, we performed source-level between-group analysis to see the interaction effects and the main effects. As you can see in the interaction effects in the first picture, in the model we kept all of our groups, our healthy controls, our overweight controls, and the anorexia nervosa patients. And we had a statistically significant result, which means that these 3 groups had a statistically significant difference in the way they process food images. That statistical significance, if you look at the second and third picture, can be explained by the difference between anorexia nervosa patients and healthy controls. Because in the second model, when we kept those 2 groups, we also got a statistically significant result. However, in the third picture, when we compared overweight controls with healthy controls, or when we compared overweight controls with anorexia nervosa patients, the results were not statistically significant. This is also reflected in the main effects, as you can see, where in the first picture, the healthy controls had a higher brain activation in high-calorie food images, which was located in the fusiform gyrus and expanding upwards towards the medial temporal lobe. However, it's quite an opposite finding here. For the anorexia nervosa patients, we identified a similar activation in the fusiform gyrus. However, the brain activation was higher for low-calorie food images. instead of high calorie. That was appeared to be the finding for the healthy controls. For the overweight controls, a different pattern appeared to be significant. However, similarly to the anorexia nervosa patients, the overweight controls, for the overweight controls, the brain activation was higher for low-calorie food images. Now, moving forward to the behavioral results, which supported, actually, the neurophysiological ones. We found that anorexia nervosa patients answered no, so which means no, not palatable, to the high-calorie images, a result which was statistically significant. As you can see that in the chart as well. This can be interpreted as a diminished behavioral evaluation of the high-calorie foods, which is supportive of food avoidance in anorexia nervosa patients. Now, to answer our final question on whether anorexia nervosa patients process food stimuli the same way they process emotionally charged general images, we followed the same methodology. We conducted within group sensory level analysis, and we identified 3 statistically significant time windows. The third time window on the right, it is the exact same time window we used for the food analysis. So we kept that intact and appeared to be statistically significant, again, in each of the groups. And we used that, and we conducted source level analysis to see the location in the brain to identify whether it is similar to the processing they made to food images. However, the results were not statistically significant, meaning that anorexia nervosa patients do not process food stimuli similarly to emotionally charged general images, which indicates possible food-specific neural modulation. This is a quick summary of everything. Food-related stimuli elicited specific neural responses in the N400, which were located in the fusiform gyrus, suggesting altered top-down semantic processing in anorexia nervosa compared to healthy controls. Also, in a behavioral level, the ratings for the palatability differed between anorexia nervosa patients and healthy controls, for high-calorie images, which show the reduced palatability of high-calorie foods in anorexia nervosa patients. Lastly, anorexia nervosa patients do not process food stimuli similarly to emotionally charged stimuli, which indicates food-specific neural modulation. That was all.
Let's move now to the last but not least, of course, presenter, Despina. Let's welcome Despina. Despina will present on behalf of Translational Neuropharmacology Lab. The title of the presentation is An Interdisciplinary Approach to Elucidate the Molecular and Neurophysiological Signatures of Misophonia. Wow, a roadmap to diagnostic biomarkers.
Hello, my name is Despina Melanthiou. I'm a PhD candidate in clinical psychology, and I am part of the Translational Neuropharmacology Lab of Dr. Zanos. Thank you very much for allowing me to talk today about my project regarding misophonia. I know that many of you might not know what misophonia is, so let me explain. So misophonia word origin comes from Greek. The one word is misos, which means hate, and the other word is phonia, which means voice or sound, and it literally means hatred of sound. So misophonia is a complex neurophysiological and behavioral condition associated with intense negative emotional reactions to specific sounds. Some sounds, these specific sounds are usually can be found in literature as misophonic sounds or triggers. And some of them are chewing, throat clearing, sniffing, and pen clicking. Some of them might be machine produced, such as a clock ticking or a hair dryer, a coffee machine, and keyboard typing. Surprisingly, though, there are some triggers that can be visual stimuli, like finger pointing and leg swinging. So, misophonia seems to affect approximately 10 to 20% of the population worldwide, and has been positively associated with high levels of anxiety, obsessive compulsive personality disorder, anorexia, autistic traits, and mood disorders. It is not yet classified disorder, and that's posing many obstacles to healthcare providers and for diagnosis and treatment. Individuals with misophonia often experience an increased activation of autonomic system, and they experience also emotional reactivity. Some of them might feel angry or have anxiety, feel disgustion, and they may experience disgust, panic, irritation, annoyance, discomfort, and distress. Also, misophonia individuals can experience body tension, muscle contraction, pain, reflexive behaviors, sweating, tachycardia, and hyperthermia, and many other responses. So to cope with stress, these individuals often have to change their behavior, and they might avoid certain places, use earphones, or withdraw from social situations. And especially if triggers involve eating sounds or repetitive noises. Overall, misophonia can affect concentration and increase distractibility, and make it difficult for someone to learn or think clearly. It usually develops in early adolescence and is possibly hereditary. Recent neuroscientific findings suggest that misophonia is linked to heightened connectivity between the auditory and the limbic systems. Also, researchers found increased activity, particularly in the right insula and the right anterior cingulated cortex, two regions that are a core part of the salience network. And this network is responsible for detecting emotionally significant information. So in the case of misophonia, it appears to attribute strong emotional meaning, specifically to misophonic sounds. Also, there is the researchers found hyperactivity in the right superior temporal cortex, a region crucial for selective auditory attention and the processing of emotional sounds. And this helps explain why individuals with misophonia might react to strongly and specifically to certain auditory cues. So, taken together, these findings indicate a distinctive pattern of brain activity that might explain both the emotional